Hi, everyone. Welcome to day 4. I am Katie Altieri. I'm a senior lecturer in the Department of Oceanography at the University of Cape Town. I'm also a member of the SOLAS Scientific Steering Committee, and I was part of the Summer School Planning Committee. So it's really nice to see everyone back for day 4. Yesterday was a really great day. It was super engaging and interesting, and I hope all of you were able to attend, but if not, of course, the recording is available on Whova if you want to go back and watch it. So today's theme is focused on SOLAS theme 4, which is interconnections between marine ecosystems, aerosols, and clouds. So we'll start with the same format that you've seen before. We'll have a couple of presentations and then a practical session. Today's practical is on science communication and the media. And then we'll finish up with the rapid fire poster presentations from all of you. I'll be chairing today. All of you, I'm sure, are familiar at this point with Whova, and that you've all had time to explore and look at each other's posters. If not during the break, you can still take a sneak peek at today's poster session. So everything's going to be recorded, and it will be available on Whova for 3 months, so don't worry about that. And of course, if you have any technical questions, Paul and Becky from Mindfully Wired are here and in the background. And you can feel free to send them a message if needs be. Their contact information should be in the chat. All right, so without further ado, we're going to have 3 really fantastic international lecturers today on theme 4. Our first speaker is Jurgita Ovadnevaite, and she's going to provide a nice high-level overview on the theme to get everyone on the same page and up to speed, because we know you guys have a variety of different background types. The second presentation will be on a research highlight. So this will be from Maria Kanakidou. And then the third, Arne Körtzinger, will present another highlight piece, but Arne will focus a little more on connecting it to some of the other themes. So note your questions. You can raise them in the chat, or you can raise your hand. But of course, as the other sessions, we will have the Q&A at the end of all the presentations. So without further ado, it will be my pleasure to introduce Jurgita Ovadnevaite from the National University of Ireland, Galway, and she'll be doing the overview talk on interconnections between marine ecosystems, aerosols, and clouds. Thanks, Jurgita.
Yeah, hello, everyone. And yeah, good morning, evening, and whatever the time is in your place. And as Katie said, that I will present the introduction to the theme. So we will be talking today about the interconnections between marine ecosystems, aerosols, and how this affects the climate through the clouds. And why it is important? Why do we look at that? And why the SOLAS is focusing and has a theme on that interconnections? Because oceans are important source of primary and secondary aerosols. And these affect direct scattering of the light, but also affect the cloud formation. And depending on the biological activity or the life in the ocean, the properties of aerosols, both primary and secondary, can differ, as many of you know, I guess, but it's just to stress more. It's just that on the right side, if you can see, the core theme 4 is focusing on the aerosols and how they form cloud or scatter the sunlight directly, and how these properties does depend on the biological activity in the ocean. The SOLAS then covers several research questions that are related to this theme. And first, it's how the aerosol load and properties are linked to the marine ecosystem. So how many aerosols, or what are physical properties of the aerosols, and also the chemical properties of the aerosols, and how they are linked to the marine ecosystems, and how does this affect their scattering and also the cloud formation. And if we skip to this and then go to the primary and marine emissions and secondary aerosols, as I said, the properties does depend on the marine ecosystems, and then they depend on how they affect the scattering and the formation of the clouds through the cloud droplets and ice crystals. But also the question is how and if anthropogenic emissions affect the natural abundance and properties of the marine aerosols. So this team also looks into this question. And what are the aerosols effects on marine clouds? Because the marine clouds are very important, especially in the remote areas, and we'll look into the effects or aerosol effects on marine clouds. And then, finally, we look also at the feedbacks between the clouds, marine ecosystems, and the climate. That's very complicated topic, but we also try to look into the feedbacks and how these interact. But why marine aerosols? Why are they important? So the oceans cover over 70% of the Earth's surface, but more importantly, that they are optically black. So I usually ask my students, what can you see in this picture? So if you can maybe quickly type in the chat as well, but while I'm speaking, what can you see in this picture? And most often I hear the answer that, yeah, we can see the ocean or sea, land, and so on. Actually, that's not true. You cannot see the ocean because you can only see something that is reflecting the light, and oceans are usually black, so they are absorbing almost all the sunlight, so we cannot see. You can only perceive the ocean because we have a land around it that reflects light, we have clouds, and you can even see the aerosols in these pictures. So anything that overlays that black surface reflects the light very, very efficiently. And from the space, as I said, oceans are black, and even optically thin layers overlaying that dark surface can have a profound effect on the radiative budget. And it's very highly sensitive system. Very small changes in aerosol availability or cloud availability can have a really high and big impact. So as I said, those white things are aerosols reflecting the sunlight, and also clouds, and they really affect the system light scattering. How do we look into the properties, or how do we understand the relationship and the feedbacks between marine ecosystems, aerosols, and clouds? So we have, of course, the long-term measurements and the stationary stations, so long-term stations looking into the marine aerosols, also interconnections with the ecosystems. We can also do the short field campaigns with the planes, ships, and also bring the ocean into the lab and larger or smaller scale experiments to reproduce the ocean. All are very important, both ambient, long-term measurements, field campaigns, and laboratory are very, very important, but they have drawbacks and advantages, and different drawbacks and advantages. For example, for the long-term measurements, you're usually on the land, so you need to take care of the anthropogenic influences to separate the marine aerosol effects from the anthropogenic influence. With the field campaigns, they're usually very short because ship trips and the planes are very expensive. So you usually have very short snapshots of the aerosol and interconnections. And sometimes that's not enough, even though you are really on the spot in the site, like looking into interactions, but they might be very, very short and not presenting the whole picture. With the laboratory, as I said, that's bringing the ocean into the lab, which is really complicated. And we could control, or we can control chemistry or physics, but it's almost impossible to predict and control the biology. They won't kind of respond to our wishes. We want maybe to reproduce something that we see in the ambient, but that's not always the case. So they do provide a lot of information or to understand the real processes behind in the lab, but it's very, very important that the laboratory experiments reproduce the real ambient, and that's usually a really huge challenge. SOLAS went even further. They tried to connect all the land stations, or the atmosphere ocean time series station, and create a network in order to better understand the ocean-atmosphere interactions, to build the capacity and share the data, and also share the experience between the different managers or scientists in different stations, and also to have that comprehensive look, because different stations would have a different information, provide different information and different small piece of the big puzzle. So combining all that information, that's the SOLAS responsibility and the huge advantage of what SOLAS does. So if we go back to the aerosols, as Katie said, I guess you have really huge background, or variety in the background. Some of you might know the aerosols or marine aerosols better than me, but some might work with the gases and would benefit from very short introduction into the aerosols. So yeah, apologies for those who are really advanced in this area, but we need to go through that introduction. So marine aerosols can originate from primary sources, which are produced by mechanical disruption. So we have that material already there, and we just produce small, tiny particles or droplets. And in the marine aerosol, that's mostly the air entrainment, so by breaking weights, these entrain the air bubbles into the water, and these bubbles raise to the surface and burst, producing film drops, if you can see in the picture. And then the cavity collapse produces the jet drops. And if we have very strong winds, they can tear the wave crest directly and produce the spume drops. So there are different primary aerosols, and they contribute to the different sizes or size ranges of the particles, but they are all primary aerosols producing via wave breaking. But then there is a really big class and also important class of secondary aerosols, because the ocean not only emits particles, there are also gases coming out of the ocean, and these, via oxidation and other processes, chemical transformations, can form particles. So we emit gases from the ocean, but these eventually would form the secondary particles. And both primary and secondary are important when we talk about the effects on climate via direct scattering of the light. For example, on the left side, you can see the haze video, or you can the obstruction of the light, and the view is not so clear because of the sea spray particles that reflect the light. And on the right side, you can see how the particles can affect the clouds. So particles can directly scatter. If you look at this picture on the left, that simplest representation, it can also result in more particles or cloud droplets in the cloud. If you look at the second from the right picture, that's called first indirect effects, because the aerosols would affect the cloud formation, but also how many droplets we have in the cloud. And increasing the number of droplets increases the scattering of the sunlight, and that's how it affects the climate. But also more droplets or more particles, cloud condensation nucleus, means that we have the same water shared among more nucleus. So it means that the droplets are smaller. And if we have more numerous and smaller droplets, it means that there will be no rain, or the rain will be farther away. And it means that there will be increase in cloud lifetime. So that's called second indirect effect. So we have both direct effect, scattering, first indirect effect, increased droplet, number of droplets in the cloud, and second indirect effect that results in the longer lifetime of the clouds. Secondary aerosols being recognized, or importance of the secondary aerosols being recognized already, and many of you have heard about the claw hypothesis. So it's not only that particles affect the clouds, but there might be a feedback between the ocean or ecosystems in the ocean and the particles or gases first emitted from the ocean, then particles forming in the atmosphere and affecting the clouds, which means that then we have higher scattering, which then should result in the cooling of the oceans and then reduction in the phytoplankton growth and then reduction in the emission of the gases. But that's not so simple. So the feedback was postulized many years ago, but the real evidence of that in the atmosphere or in the ambient was not really proven. So we kind of proved some small parts of that feedback. Like, for example, in this study, that they showed that increase in the nanophytoplankton cells in the water results in the increase in particle numbers emitted from the ocean. So we have a direct link between the systems of the phytoplankton in the water and the number of particles that is emitted from the water. So we have that link. However, how these particles further affect the clouds and whether that affects the water temperature and phytoplankton, that link is even more complicated to capture. Seems there's a problem to advance some slides. Okay, it was maybe too big. The picture was too large and it was taking. So that link between the phytoplanktons and particle number has been shown in several studies. So it's the higher productivity or higher amount of the algae, microplankton in the water results in the difference of the water chemistry, but also that affects the particles emitted, both primary and secondary, but we are talking now about the secondary particles. And these particles can be very efficient cloud condensation nucleus. So the previous study just showed the link between the ecosystems and the number of the particles. Here on the left right side, you can see that these red dots, they represent the cloud condensation nucleus arising from these secondary particles. And it seems that the measured CCN are much more abundant than we could predict with the Kappa color. And that means that they are really efficient cloud condensation nucleus. Okay. And if we, from this, just very briefly, in this example, that when we look at the composition of these small particles or secondary particles, they can both be organics and sulfate, and therefore the Kappa color struggles to represent these, and we need more sophisticated theories in order to understand the cloud activation of these particles. We won't go into details. If you want, you can just look at these papers to understand more scientific details, but the thing is that the organics from the marine ecosystems as well as sulfate plays a role in the cloud activation. And there are other many studies showing that link between ecosystems or phytoplankton blooms in the water and the particle or new particle, secondary particle formation. Okay, apologies for the delay. Yeah. But it's not only the secondary particles that are important. Previously, primary sea spray was kind of dismissed because it was assumed that the sea spray only contributes to large particles. So we have only supermicron sea spray, and small particles do not play a role. Actually, new studies from both laboratory and field experiments, we see that primary sea spray particles can be as small as 10 to 15 nanometers in size, so they can contribute to the Aitken and even nucleation mode, and therefore can have an effect on clouds. And it really depends on the method, how you look. For example, in this paper, they just assume that the sea spray is super micron. And unfortunately, I cannot point you with a pointer, but if you look at the graph, so the blue is the sea spray, and they just assume that it's super micron. And if you assume that large particles, then yes, of course, they do not contribute much, neither to the scattering nor to the cloud formation of the the clouds. But if you look in more details, we can see that the sea spray can also be as small as I mentioned, as 10 to 15 nanometers in size. And if you account and look with the direct methods, so in this paper, they measured particles, small sea spray particles with direct methods, and proved that they can be in the Aitken size. And then if you account for that, then the sea spray can result into 500% more nucleus than with just supermicron method alone. So my take home message from this is that depending on the method that you determine the sea spray, the results on the effects of the sea spray can differ significantly. So that's really important to remember. And when you talk about the sea spray, or when you talk about the sea spray effect, is how you determine what sea spray is and which size ranges it does cover. Okay. Yeah. And sea spray is not only sea salt, so the part of sea spray can be organics, and that the composition and the chemical composition really affects how the sea spray can affect the direct sunlight scattering, but also the cloud formation. And in this picture on the right, you can see that in the winter, the top graph, we don't have much of the organics, and chlorophyll activity is low, and the green in this case is organics. But on the bottom, during summertime, when the chlorophyll activity or biological activity is high and we have abundant chlorophyll, we see that the particles, sea spray particles, can be composed of organics. The smaller the particles, the more organics contributing to the composition. So we just go back again how the sea spray is formed. So we have those breaking waves and training the bubbles into the water. When these rise, they can scavenge the organics there in the water. And then when the bubbles burst, part of the produced sea spray aerosol can be coming from the organics from the water, as well as the microlayer on the top. So it's not only the sea salt that composes the sea spray, but we can have organics. And as I said, that hugely affects how the sea spray then can scatter the light. On this graph, it just shows how the scattering efficiency reduces if we go from the pure sea spray or sea salt, the high hygroscopicity. So it means that if you have sea salt, the particles are water attractive, so they love water, they can grow large droplets and they can scatter light very efficiently. But if you add organics, which is hydrophobic, it means that it's hating water and the droplets cannot grow as big, so the light scattering will be diminished significantly. Also, organics can affect the cloud formation. On the right graph, you can see that the same hygroscopicity, like reduction in hygroscopicity, arising from the organic contribution. So red bubbles here showing the high contribution from the marine organics. But we see that even though the growth factor is very low, so they would not contribute to the scattering much, these are very, very efficient cloud condensation nucleus, because the ratio between CCN and the real cloud nucleus is almost 1. So the composition of the particles affect both the direct scattering, which is diminished, but they can promote the cloud formation of the marine particles. And why it is important to understand these effects, because in the modeling, like in the understanding of the climate effects and how sea spray affects the scattering, it's really important to understand the properties, because if you just look at the sea salt, the direct effect of the sea salt is negative because we have the scattering, however, indirect effect is the positive. I won't go into details in that because if you want, you can look at the papers, like the larger particles would diminish the scattering by the clouds, or larger droplets, darker droplets. But the important thing from this is that if we add the marine organics, as I mentioned before, that would affect the overall sea spray effect from being the negative or positive in the indirect to being negative indirect effect if we add organics. So again, you don't need to remember those details, but the composition of the sea spray particles affect the overall direct or indirect effects of the particles in the model. So we really have to understand these and represent correctly the correct size ranges, but also the chemical composition in the models in order to understand their global effects. And how do we relate? Because we cannot just, because the marine ecosystems are very complicated, so it's very difficult to really physically reproduce all the ecosystems in the water. So we sometimes use the proxies. And even today, the best proxy to reproduce the abundance of the life in the water is to look at the chlorophyll. So to relate that organic enrichment or how much organics we have in the particles, we can relate to the abundance of the chlorophyll. Again, it's not so simple because there are other factors at play that can affect that chlorophyll, and the link between chlorophyll and organic enrichment is not so straightforward. But even more sophisticated proxies like net primary productivity, even though increases the correlation between the enrichment and the abundance of the, or the value of net primary productivity versus chlorophyll, it's still not ideal proxy to understand all the effects on the particle composition. So as I said, it does improve somehow, but the even more important factor actually is the time resolution. So if you go on the ship and you sample chlorophyll and you try to relate that instantaneous chlorophyll to particle composition, that would not work. Like you need maybe longer time trends or longer periods in order to have that better relationship between the chlorophyll and the particle composition, because the instantaneous chlorophyll might not represent the chlorophyll that was affecting the particles during the formation itself, because the particles that you measure on the ship, for example, can be formed far away with different chlorophyll properties than you do measure from the ship. And again, the picture is even more complicated. Like it's not only the understanding the secondary particles forming from the ocean, primary particles coming from the ocean, and their physical or physical-chemical properties. No, there is also interaction between primary and secondary particles. Like in this example, for example, we have the activated sea salt particles can reduce the cloud supersaturation. Like on the left side, if you look at the picture, you see that the cloud peak supersaturation reduces with activated sea salt, because the sea salt attains the water in the cloud, and it means that less water left, or the lowest supersaturation would be for the secondary particles, and it means that we would have fewer secondary particles activated in the same cloud. So as I said, we cannot just look separately into primary or secondary sea spray. We have to look into the complex system if we want to understand how these particles affect the cloud formation. And if I have a few more minutes, that maybe I'll just present some aspects of the ice nucleating particles. So before I was talking mostly about the cloud condensation nucleus, but ice nucleating particles are as important, if not more important, especially in remote areas, even though the abundance is very low, but the importance in remote southern oceans is very huge. But understanding of the particles at the right side is quite low, especially of the ice nuclei or particles that are originating from the biological activity or from the organics. We have very low understanding in the parameterizations, but also in the aerosol modeling. They are not properly reproduced, therefore we struggle to properly have their effects in the models. Yeah, apologies for delays in slides. Yeah, and it's even more complicated that it's not only like with the CCN, we could maybe relate the organic enrichment and the cloud properties to the chlorophyll. With the INPs or ice nucleating particles, you might even need to have complicated or dual parameterizations in order to capture the properties of the particles. And it's also, as I mentioned, the lab experiments should reproduce the real system. And actually, the ice nucleating particles, it's even more complicated because the particle generation method matters, and different lab or chamber experiments might not be really representative of the ambient. Similarly to the CCN systems, as I said, bringing the ocean into the lab is complicated, and it's very difficult to capture all the aspects. And that's it. With this, I'll finish my talk, and yeah, I'm happy to accept all questions, maybe either now or after the session.
Thank you so much, Jurgita. Wow, that was a fantastic synthesis of the literature, recent literature, and big papers in the field. I think that'll be a really great resource for the students. So thank you very much for that. Next up, without further ado, we'll move on to our next talk by Maria Kanakidou from the University of Crete. She's going to be sharing a research highlight with us entitled Atmospheric Cycle of Aerosols in the Marine Environment. Maria, you can unmute. It's all you.
Thank you very much. It's a pleasure to speak after Jurgita. She explained everything. A really great highlight of everything. So I will just try to go on and provide some things that I consider are interesting in this field regarding aerosol in the marine environment. We all know that aerosols in the marine atmosphere are not only coming from the ocean itself or the region there, but they come also from terrestrial. They have also terrestrial origin, and they have a number of impacts. They can reduce radiation that reaches surface. They can affect the cloud properties. They can form warm clouds or ice ice clouds or mixed phase clouds. They can carry also nutrients like phosphorus, iron, and nitrogen, copper, etc. And carrying those from land to the ocean or from ocean to land also. They can release them to the ecosystems and affect ecosystems. So thereby the functioning of the marine environment, the marine ecosystems is disturbed by the presence of the aerosols in the marine environment. And there just to mention that when we talk about function of marine ecosystem, we look on the source of atmospheric oxygen, which is 50% of the oxygen we breathe roughly comes from the photosynthesis into the ocean, and also the sink of carbon dioxide. But then in areas that are anoxic, then we have a source of nitrous oxide, which comes to the atmosphere, and it's also greenhouse gas. So it's aerosol can play an important role in the marine environment. the most well-known gas phase precursor of aerosols coming from the ocean is the dimethyl sulfide, which is oxidized into the marine environment by different, mainly most of the oxidants present in the marine environment, like OH radical, ozone, and halogens, oxides of halogen, BRO, and chloride also. And this chemistry can happen in the gas phase, or can happen into the liquid phase, in droplets, or in the water of aerosols, and can form, as you see here, from DMS in the middle. You have major products like sulfur dioxide, which bottom left, which is known for its anthropogenic emissions also, but in the marine environment it can come quite a lot from the oxidation of DMS, and this can form sulfate, bottom right, and the two major products coming out from DMS oxidation in the atmosphere is sulfate and methyl sulfonic acid, MSA, and those are going to the aerosol phase. So this paper I put on here from Chen and co-workers has done a global modeling study and found out that DMS is mainly oxidized in the gas phase by OH radicals by 66%, and then nitrate radical, which is mainly of anthropogenic origin, is contributing also by 16%, and then the halogens, mainly BRO, by 12% on a global scale, and this is mostly over oceanic regions or coastal regions to have the nitrate radical. So they also found out that MSA has an important contribution in terms of production in the marine environment by aqueous phase reactions that involve OH radical and ozone in cloud droplets and aerosols. What is important here is that those compounds have different lifetimes, and what I have put in red spots, red circles, not rectangles, are those that have the longest lifetime. MSA can be present in the atmosphere for 2.2 days, where sulfate, according to this modeling, is about 5 days, 4.5 days. So those we can measure them, and we can find that they are very affecting the marine environment. This is, however, the basis of what was known before recent years. This is the latest in 2018, knowledge in 2018. And on this slide, we can see this, the chemical sources of MSA. The top two panels show the production in the aqueous phase from OH radical to the left and from ozone reactions to the right. And the bottom shows similar oxidants, the OH radical in the gas phase, and the ozone reaction in the gas phase to the right. So what we can see, those are percentage contribution to the total source. And why I show you this one is that to tell you that different processes can be important in different regions. So it's not the DMS oxidation, it's not governed by one chemical oxidant, but by several of them. And on this slide, you can see the distribution of the oxidants, how the slide, now it's difficult to show that. Top right, you can see the chloride distribution, and next to it, to the left, you can see the BRO distribution. They're quite different in both in levels, but also in in the distribution. And at the bottom, you can see the ozone distribution by their model and the OH radicals distribution. So it's important to take into account all chemical reactions or processes, major processes that can affect DMS oxidation. And this is not the full truth, because recently it has been found that one compound, this hydroperoxymethyl trioformate, that is shown at in the middle at the right top, is also a very important compound. It has been measured into the atmosphere, and when we account for this one, we can better simulate what happens into the environment. So this has been measured during the Atom 3 and 4 campaigns, aircraft campaigns. On this slide to the left, you can see the cruises of the aircraft, and you can also see on a grayscale you have the DMS levels into concentrations into the sea, and with dots on the cruise track you can see the concentrations of the HPM MTF in colors, and the red is the highest and the blue is the smallest, and these measurements happened also in September 2017 and April-May 2018, so two different periods. And to the right hand you can see the measurements on the vertical. This compound is very abundant near in the boundary layer, and it's reduced when we go high up, but it's there in significant amounts. The vertical profiles totally to the right show the comparison of the concentrations of DMS with green triangles, SO2, which is the well-known product of DMS oxidation, with lighter green squares, and this new compound, the HPMTF, with red color. So this has been identified in the field and has been also generated in the laboratory to prove that indeed this compound, and by the observations we can see that when we have clouds around the blue regions here, then we have a relatively low concentration of HPMTF, which shows that it's fastly removed into the clouds. A very interesting result with regard to the aerosols is shown on this figure, that shows that at the top colored plot shows the aerosol number size distribution. So the colors show the number concentration of the aerosols, and the y-axis shows the diameter of the aerosols. So if low down, where we have the 10, for instance, correspond 10 nanometers of aerosol diameter, which shows practically the very small particles into the atmosphere. And the bottom panel shows this the variability of this compound, the HPMTF compound with a black line, and the variability of the aerosol number concentration for the particles that have sizes smaller than 10 nanometers. That means for the particles that are really part of what we call new particles formed into that environment. So we see a very strong relation between the DMS chemistry practically and the new particle formation into this experiment. the outcome of this study is that DMS contributes to new particle formation, but it's not the only one. So we need, if we see what happens during summer in the Arctic Ocean, then we find that there is a lot of ammonium sulfate, which is the most known, I would say, aerosol precursor. And this is, however, found to be, it has been found also to be coated by organics in this environment at the relatively high relative humidity, where we expect this one to be liquid, but it is found in solid phase, and about 30% of the particles below 0.2 micrometers in terms of number concentration, they were found to be ammonium sulfate coated by organics and be solid. So this has been explained by the impact in reality of marine biogenic components, and also the fact that a particle collision will result in solidification through contact efflorescence. That means going from liquid back to the solid. And those solid particles could nicely contribute not only to cloud condensation nuclei, but also to ice nuclei. And there we need to say rapidly few things. The cloud condensation nuclei will grow up and will form warm cloud droplets, while the ice nuclei will end up to form finally ice in the clouds and contribute to ice clouds or mixed phase clouds. And as ice nuclei particles, we know that bacteria can act like that. We know also that the dust can act like that. Pollen and fungal spores can have this ice formation property. So you can just go to this paper and find more about it. It's a very nice review on ice nuclei particles. I would like to spend a few minutes that are remaining on showing you one of the first studies that looked on the effect of aerosols in cloud formation, shallow cloud, warm clouds also formation via satellite observations. And this was from Kaufman, a very well-known scientist who passed away suddenly. And what he has done is, and his co-workers, they have looked the satellite observations from ODIS, and they have shared the observations in three zones: the one where you can have mainly pollution aerosol, the one where you can have most dust aerosol, then the smoke aerosol, biomass burning in the South Hemisphere tropical zone, and then the sea salt. And they compare that with the cloud cover from again from satellite observations, and then they studied how much of those aerosol can affect the shallow clouds in green here, or convective clouds in red here. And to make it shorter, they found that they found a significant radiative effect at the top of the atmosphere coming from clouds, and both accounting from the shallow clouds, their impact on shallow clouds, and also from the impact of the direct effect, the impact of solar radiation. And about 2/3 of it was due to the aerosol clouds interactions that Jurgita explained also, and 1/3 was due to aerosol direct radiative effect. And they also evaluated that they have a progressively increase in size in cloud size droplets when you go from clean to polluted, smoky, and then dusty conditions. They have been able also to calculate the top of the atmosphere forcing of shallow clouds, and here you see roughly it's in watts per square meter for the period of the short months period they studied. You can see where this impact is seems to be the highest. So one more thing I would like to mention is about the share between the radiative forcing due to aerosol cloud interaction. That means how the aerosols make the cloud brighter, and they change the forcing and the cloud adjustments. That means how the aerosol change the height of the clouds. And there is a recent study looking on the warm marine clouds, combining satellite observations from the Nasser train and model results. And they find that the top figure here, we can see the radiative forcing due to aerosol cloud interactions due to this cloud brightening. And this is 80% of the total due to aerosols in the marine environment. While the cloud adjustment, that means the change in the height of the clouds, contributes only to 20% of it. So I don't know if I can continue or I have to stop here. So I have to stop here.
Yep, I think we should wrap it up, Maria.
Yeah, give me one more slide, then this will be about the ice nucleating particles, which are very important, and they are very important in the marine environment. We have seen already, Jurgita mentioned this, but I would like to show you why they are important. If you have a liquid cloud, a supercooled liquid cloud, then you have a highly reflecting system, and you have less light going to the surface. Once you go in the middle situation here, where you have some aerosols that can be of marine origin, or they can come from dust, or can be terrestrial biogenic particles that are blowing with wind over this area. Then you start having ice nucleation. So the liquid is reduced, and ice cubes, ice crystals are formed into the cloud. So we go to the right side case, where we have the shortwave radiation, we have less reflected because the cloud becomes more transparent, and then more light gets into reaching the surface. So this is important, and it has been found that large biases in climate models might be due exactly to the wrong representation of ice crystal in the models, and the marine environment is very important in this respect. So I will stop there, and thank you for your attention. And questions will come, I imagine, at the end of.
That's great. Thank you so much, Maria. You really went deep on a couple of topics that Jurgita introduced, which is fantastic. All right, next up, we have Arne Körtzinger from GeoMar, who's going to talk to us a little bit about eastern boundary upwelling systems, and we'll make some connections to the other themes. So thank you very much, Arne. It's all yours.
Yeah, thank you, Katie, and welcome to everyone. So I'll be talking about upwelling in the ocean, which is a widespread phenomenon, and it can range from short-lived localized upwelling like at island tips or in coastal areas to seasonal or even perennial large-scale upwelling, such as in the eastern boundary upwelling systems. And I'm going to focus now in this talk entirely on the eastern boundary upwelling systems, which arguably are the most important upwelling phenomenon in the ocean. They clearly are of huge socioeconomic importance, and I think they are wonderful hotspots of solar science, as they actually map on all 5 core themes, including, of course, the aerosol story. So with having said that, I'll go into it, and it's going to be a little bit of a tour de force through the science around upwelling systems. And I start with some physical basics before I guide you on to the biogeochemical and biological consequences of upwelling. So upwelling is a wind-driven phenomenon. The reason for it is the existence of those large high-pressure systems in the subtropical regions, the subtropical highs on top of or above the subtropical gyres. And then on the eastern side of the basins, we typically have hot and dry, specifically during summer, areas where low pressure systems develop. So we have a relatively large pressure gradient between these water-based highs and the land-based lows. And the isobars between these pressure systems in certain areas are parallel to the coast, such that the winds, and these are of course the trade winds, run are pretty intense, and they run parallel to the coast. Because of, and I would like to say that there are these 4 major eastern boundary upwelling systems, and you probably know all of them. There is a similar system we call not the eastern boundary upwelling system, but it's a wind-driven system that shares some similarities. That's the monsoon-driven system, the Somali current system in the Arabian Sea. So that's comparable in many aspects. Okay, so we have this situation in the atmospheric pressure systems, which give rise to the trade winds, which run along the coast because of Ekman theory. This drives an Ekman transport of water away from the coast into the open ocean. So because you push water away from the coast, the sea level will be lowered right at the coast. So we have an inclined sea level towards the coast. This in itself drives or gives rise to a pressure gradient, and the pressure gradient induces a geostrophic flow which goes equatorward. So we have a superposition of wind-driven Ekman transport and equatorward geostrophic flow, which really pushes a net transfer of water away from the coast. Of course, that water for hydraulic reasons has to be replaced from underneath. And that is exactly the upwelling we're talking about. So the wind-driven Ekman layer pushes the water out. It's replaced from underneath. Depth from which these waters originate are on the order of 50 to 200 meters. And that water is supplied typically across the shelf in a bottom layer from the open ocean. And so there has to be a supply mechanism of that water from the open ocean because of complex physical reasons. There is also a meridional pressure gradient, which drives what we call the polar undercurrent. That's a poleward jet-like current, typically at the shelf break, and that undercurrent is actually feeding a lot of water into the upwelling system. And that's important, as we will later see, because even in the northern hemispheres, the waters that are advected through this poleward undercurrent originate mostly from the southern hemisphere. So we have far-reaching influences onto this relatively small local phenomenon. Okay, so we have this upwelling driven in the region. It brings up waters from subsurface depth, 50 to 200 meters, and the most immediate effects of this is, of course, the waters are colder. So we see it clearly in sea surface temperature. And in this talk, I'm going to be mostly focusing on the Northwest African system, the Canary Current upwelling system. You see the vectors of the trade winds. You see that there's a seam of relatively or sometimes very cold water that upwells along the coast. This water, of course, is not just cold, and there it becomes important for biology and biogeochemistry. It's full of nutrients, and that is largely because the waters that are advected with this poleward undercurrent originate mostly in the southern hemisphere. They include mostly South Atlantic central water, which is produced actually at the poleward side of the subtropical gyre in the South Atlantic. And this water has traveled across the South Atlantic, across the entire tropical ocean from west to east, before it slowly moves into this area. And during this passage, it has lost a lot of oxygen through consumption and has accumulated nitrate. So actually, if you look at the distribution of nitrate at 60-meter depth, so around the source depth of the upwelling, you see that there's an elevated level of nitrate because of the feeding of nitrate-rich waters through this poleward undercurrent into the system. So there's far-reaching influences from the southern hemisphere that play out in this region. Now, not surprisingly, if there's a supply pathway through the upwelling of nutrients to the surface, that induces primary productivity. So from space or even at the sea, of course, if you go there specifically during the productive period, so you see strongly elevated levels of chlorophyll, really high concentrations of chlorophyll A. And before we go into looking a little bit more into the specifics around the biological productivity, I would like to bring in another aspect that, as you will see, is important for biogeochemistry and in the end SOLAS science in the region. And that has to do with the fact that the region, these 4 eastern boundary upwelling systems are not just home to upwelling and upwelling induced productivity, but they are also home to oxygen minimum zones. And the reason for this is the fact that oceanographers call this shadow zones. So these are regions which are kind of left out the typical ventilation schemes of the ocean. So neither from the north, where we have the subtropical gyre, a lot of freshly ventilated water is advected into the shadow zone because there's a Cabo Verde frontal zone in this case. You have frontal zones that, allow waters not to penetrate into the shadow zone from the poleward side. And then also we have the vigorous staggered jet system in the equatorial region with eastward and westward going jets. But these jets, which are reasonably well ventilated, don't connect with the shadow zone from the southern side either. So these shadow zones are really not ventilated at all. They have very poor ventilation. And as even the ones in the northern hemisphere are ventilated from the south, the source waters have very remote outcrops, have reasonably long travel times, order half a century or so, or even longer. So they have lost a lot of oxygen on the way. And this is one more reason why we have oxygen minimum zones in the region. So we have the sluggish supply of oxygen to these shadow zones. And at the same time, these shadow zones are in the vicinity and in the influence of these high productivity areas, of course, which drive high biological productivity. A lot of the biogenically produced carbon is exported vertically and laterally into this region. And we find much enhanced oxygen utilization rates in the interior ocean, particularly in these areas where oxygen is supplied very sluggishly. So these regions are a superposition of high oxygen demand and low oxygen supply, and then you end up with a low oxygen minimum zone. Actually, the one in the North Atlantic is the least one developed, and the ones in the Pacific are much more massive and important. So we have this additional component of marine biogeochemical forcing from underneath. So let's see how that all plays out. We've seen that all 4 major eastern boundary upwelling systems have this hugely elevated chlorophyll concentrations and productivity. And they all are kind of different, and the forcing factors behind it are different, but they are similar in that relatively small areas of the ocean are home to a huge fraction of the productivity. So they give rise really to a brimful ecosystem, and it's not just brimful in a biomass sense, but from a biological perspective, you will find out that, and that's shown in the bottom left figure, which shows an indicator for species richness in oceanic taxa, so how biodiverse is the ecosystem in the open ocean. And you see that some of these ecosystems, particularly one of West Africa, has red colors, so it's on the absolute high side in terms of species richness, on par with really the famous Cape region or the Barrier Reef. So we not only have huge productivity, but we have very biodiverse ecosystems. Of course, you all know that these regions are home to the most intense fisheries. Unfortunately, some of these regions are also home to the least well-managed fisheries. So there is huge socioeconomic effects and anthropogenic forcing factors that also coincide in the region, and we have no time to go into this. Now, when you look at the productivity in the ocean, it can be calculated as in a simple equation, including biomass, the maximum productivity rate, and then limiting factors. And you can easily draw a scheme like this, which shows how all the different physical and biogeochemical factors around it will have an impact and will shape the productivity. And there is physical things that transport, of course, the cost, the support of the nutrients, but also are involved in the fate of the produced material, the subduction of nutrients, and so on. So it's a complex situation. And of course, limiting factors of primary production can be from light, and that connects, of course, to the cloud topic we've heard before, but also from nutrients. And it's not just the major nutrients that are supplied from underneath that play a role, but also micronutrients such as iron. And that's another beautiful connection to what we've heard in the previous two talks. And let me just show you that this is one of the areas where you would exactly look for some of the things Maria and Yogita have talked about, where we know that a lot of, it's actually the dustiest place in the ocean of West Africa, you can almost say, where a lot of material is provided through the atmosphere. Dust is delivered to the ocean, limiting nutrients such as iron, cadmium can be released through it. And that is a very important link, sort of a forcing from the atmosphere on the primary production in the ocean. And that has been highlighted already today. But then we have this oxygen minimum zone underneath, and that also provides an important forcing on nutrients, because the redox conditions in the ocean have a lot of bearing on nitrogen or nutrient cycle in general, but also nitrogen cycling in particular. It's because under certain low levels of oxygen, below certain thresholds, microbial pathways, metabolisms kick in, such as denitrification, anamox, and different reactions that impact heavily on the availability of nitrogen, the bioavailability of nitrogen, but also in the contact with the sediments. We can have under low oxygen conditions, ammonia, iron, phosphate releases into the waters that, provide the source of the upwelling waters. So these low oxygen conditions really shape the availability of nutrients, the stoichiometry of nutrients, and thereby shape both the quantity and the quality of the biological production from underneath. So you really have from above and below forcing on nutrients and their stoichiometry, which make this, you can call it complex or interesting, but it makes it very dynamic. Now, as we've seen in some of these oxygen minimum zone, we know that under anthropogenic forcing, warming mostly, these oxygen minimum zones, some at least, tend to both intensify and expand. So there is good reason to believe that oxygen forcing from underneath will intensify. And that's not only true for this kind of static oxygen minimum zone, but we can even show that the eddies, which are also important in transporting biologically produced matter away into the ocean, into the open ocean, can even develop short-lived but pretty drastic oxygen minimum zones themselves. So we found these anticyclonic meltwater eddies of West Africa, within the core of which we really have dead zones, so extremely oxygen poor waters that even start to become denitrifying. So it's not just the big picture that matters, but even the smaller scale, the mesoscale, even the submesoscale, we call it in the ocean, matters for production and the fate of the biologically produced matter. A complex situation that warrants research, of course. And of course, the waters that upwell not just bring nutrients to the surface, macro or micronutrients, but a lot of greenhouse gases. Upwelled waters have been shown to be rich in CO2, often elevated in methane, very often very rich in nitrous oxide. So a lot of greenhouse gases really under the good trade wind forcing outgas from these regions into the atmosphere. But then for CO2, of course, the upwelled CO2 will be quickly also consumed again through the nutrients which are there. So for CO2, the overall balance really depends on the stoichiometry of the nutrients to carbon as they are upwelled, and then on the stoichiometry of the produced biomass and the fate of the biomass and the upwelled nutrients. So it's a complex thing, but not just radiatively active trace gases emitted, but also a whole range of chemically active trace gases chemically in the atmosphere. We have heard about oxygenated compounds, about unsaturated compounds today, about halogenated compounds. And I'm just showing the example of bromoform and dibromomethane as examples of substances which actually are at elevated concentration and hence fluxes. Isoprene, DMS, all of which, or precursors of DMS, all of which have been shown to occur sometimes at very elevated concentration. So the whole gas story is present around these upwelling systems, but not just on the production side. Also, if you look closely into the mechanics of the air-sea exchange itself and how we are parameterizing it. So since and this will come a bit later, but since there's a lot of organics in the surface of the ocean, the effect of surfactants on the trace, on the gas transfer coefficient is something you want to study, may want to study in this area specifically. Now, zooming in at the surface microlayer, and that's been done luckily in the talks before, you see that there's a whole, I would call it a microcosm of processes in the upper parts of the ocean, and there's different layers that people have identified, whether the micro layer or the thin film or even nano layers at the top of the ocean. But it's very obviously that a lot of processes are kind of focused or concentrated in this area. This is both, or in this layer, these layers. It's not. This is both an area, is an habitat for organisms. So you see production and processing of organic carbon, of gases in that very layer. But at the same time, a lot of material is released to the ocean, and that layer is then also if you want to look at it as a UV chemistry or photochemistry reactive layer, where reprocessing, oxidation, production of unsaturated molecules can take place in that kind of focused UV exposed layer. Particles, living or dead particles, interact with solvents, with polar, non-polar substances. So a lot of interesting chemistry is going on there, which has bearings, of course, as we've heard, for the production of aerosols, organic aerosols, coatings to the sea spray that is expelled from the ocean. But also, as I said earlier, it has bearings for how we parameterize or estimate that gas transfer coefficient in the region. And these regions have been shown to have high surfactant concentrations, even slicks at low wind speeds. So there is additional resistance to gas transfer, and also even the production possibly of gases in that layer. A lot of chemistry going on there. And on top of this, it's the recognition that the major forcing factors for the global ocean, which is warming ocean acidification and oxygenation, all are superimposed in these eastern boundary systems. So all of these interact at the same time. And this gives really rise to the question how much synergistic or antagonistic effects on the ecosystem or in marine biogeochemistry may be at work in this place. Bottom line is, I think these are wonderfully connected and complex systems, and rightly, they have been identified at hotspots of SOLAS science. And I think that's, my time is up anyway. I end up with this advertising, if you will, these eastern boundary upwelling system as wonderful test beds of our understanding, particularly of the connectivity between physics, chemistry, and biology in systems that really matter also from a human perspective. Thanks for your attention.
Thanks so much, Arne. That was fantastic. Upwelling is such an ideal example of a cross-cutting theme of SOLAS, and I think you really touched on that really nicely in a bunch of different ways. So thank you to all three of our speakers. Up next, we have our question and answer session. So now I'd really like to encourage you to turn your camera on if you can, if you have the bandwidth, and if you feel comfortable doing so, just because it'll help us to see your face and see some smiling people in the photos. You can feel free to ask a question using the chat feature here in Zoom, or you can raise your hand, in which case we'll call on you and ask you to say your question. If you put your question in the chat, I'm happy to read it out if you'd like me to, or I may ask you to go ahead and do that, depending on how you see fit. We've lost your Gita briefly, so we'll focus in the beginning on questions to Maria and Arne. And just before we start, I thought really the three talks were together a really nice tale of the connection between physics, chemistry, and biology. Yogita and Maria started off by talking about the biology in the ocean, influencing the chemistry in the atmosphere, influencing the physics of clouds and radiation. And Arne took us right back to the flip side, where the physics of the global ocean atmosphere system are influencing the chemistry of the nutrients, which is influencing the biology of the surface ocean. And then we've got ourselves in just a lovely ocean atmosphere loop, which I thought was really fantastic. So there were a couple of things that I think, just to get us started, I want to ask both of you to talk a little bit about scales. It's one of the things when we think about this topic of clouds, climate, things like upwelling, we're talking about things on a massive global scale, but yet you both took the time to really focus in on super small scale, whether it be if it's a liquid inside an aerosol shell, or a solid inside an aerosol shell in Maria's example, or Arne, as you mentioned, upwelling is this big global phenomenon, yet you took the time to focus in on the micro layer. Would maybe each of you comment a little bit about how you think about scales in the context of your particular research topic, and why it's so important to be able to go from the really big to the really small?
You want to give it a start, Maria? Shall I go? Whatever.
Doesn't matter. I can go first, short this time. Okay. I think what we try to understand on a global scale depends on processes. And the processes are not on global scale, they are on small scale. So the challenges we have now in, at least my field, the atmospheric chemistry, as I see that, is to understand what happens in the small scale and be able to convert it, to transfer it to the larger scale. So it becomes a logo now, from molecules to the globe, something like that. So we need really to understand the small scale impacts and convert them to the large scale. The large scale has the, that means what we see on a molecular scale will definitely change, because in the atmosphere we have the mixing. And the atmospheric transport will mix our compounds with something else. In the slides, I did not manage to show, for instance, the natural components with acidity, pollution components, which will give a different result. So we need to understand both what happens on the molecular scale and see how the mixing into the environment will lead to a global product. So,
Yeah, we keep to say that all scale matters, which almost sometimes sounds like a selling point or a salesman's perspective, but I think it's true, and both from a spatial as well as from a temporal scale perspective. I think I tried to show that from the global scale, where water masses produced in another hemispheres really influence chemistry at regional level, down to indeed the molecular scale when it comes to processing of particles in the surface microlayer or nanolayers of surfactants. This all matters, and there's interesting scales in between. So it's not just the global and the molecular scale. The eddies, for example, that I referred to are responsible for a significant fraction of the productivity in these regions, and also for the export, lateral and vertical export. I mean, it's hard to quantify it, and actually we're after quantifying it, but we know that these eddies are important features, and they kind of mesoscale, so 100 kilometers across. But once you look at these in more detail, you realize it is actually certain features on these eddies, submesoscale, kilometer scale, even below that, that drive the physics. So it's not the whole thing, rotating thing that makes things act, but it's parts in the eddies where have divergence or convergence, which drive things and which produces export. So the typical way things go is the closer you look, the more attention you realize you have to pay to smaller scales, and that goes all the way from the global to the molecular scale. And it's not just a salesman argument. I think we can make strong cases that these different scales, also the ones in between, do really affect the overall functioning of the system. You can call this a problematic situation, but from a science program perspective, it's a wonderful situation.
Yeah, absolutely. The more complexity, the more it keeps us busy, and lots of scientific questions to ask, which is great. So we have a couple questions from students. Hashan, would you like to start us off? I see your hand came up first.
Yeah, my question is to Ani. And you were saying like anthropogenic global warming increases the land to ocean pressure gradient with the temperature gradients. So it means that the upwelling intensity will increases. Will it increase? Yeah. So yeah. So my question is like, how does it affect the duration of the upwelling? I mean, we know that the intensity will be high or increases, and how about the duration?
Yeah, understood. So I think even the first point is not so clear. The first point that you made is not so clear. Of course, the simple argument goes that the intensification of the pressure gradient and the corresponding intensification of the trade winds should drive more upwelling. An analysis of the data do kind of show this in some upwelling systems, but in others we have very conflicting evidence going both ways. What we more clearly see is a poleward shift of upwelling cells. So I think the jury is still out on where we find an intensification and where we find a relaxation and how this works out. It's this old more simple Bakun theory may actually work in some places, but it doesn't necessarily work in other places. And I think that in itself is probably a complex thing to answer, whether the duration of the upwelling, of course, in some regions it's only a seasonal phenomenon, Arabian Sea anyway, but also in the big upwelling systems, they usually have a perennial part and the seasonal part. And of course, also these parts may shift poleward. So I think it's probably not a simple answer to something that seems simple on the theory side.
And I have another question. So are all upwelling systems act as CO2 sinks or CO2, degassing?
This is also something that sounds easy. Of course, the upwelled water are rich in CO2 everywhere because they are kind of older water, which have a huge respiratory CO2 imprint. So they all are rich in CO2, and among the CO2 richest water you find anywhere in the world ocean. But if you do, which we can't do easily, the full integral over of the fate of all the upwelled water, and when it is then advected out in the ocean, and what is the biological consequence that occur on their way out. If you do the kind of the bottom line across everything, it's not even clear they are a net source. Probably they are, but it's far less than what you would estimate if you just look at the upwelling cell itself, where you have huge outgassing. So even that balance for the CO2 we cannot cast easily, and that in itself is a weakness in our understanding of the overall effect of these ecosystems. Thanks.
Thanks. 2 great questions there. We have another hand up here, Lee. And would you mind, sorry, I should have said this before, but maybe just introducing yourself and saying where you're from before you ask your question?
Sure. Everybody, my name is Lei. I'm a postdoc at SUNY ESF. I have a very general question about the first talk, if the speaker is still here. Probably it doesn't matter. So does anybody ever look at the chlorophyll relationship with I remember that plot with showing the chlorophyll with the number of aerosol or the number of aerosol. Does anybody ever look at the relation in oligotrophic ocean? See if that correlation holds or not. Since there's I think the majority of the ocean is oligotrophic, right? How valid using that chlorophyll as a proxy to study the aerosol, because we know there's a large carbon background in the ocean, right? It's about 650, in the surface ocean, you see it's about 50, 60, 70 micromolar. So, when it's oligotrophic, how much organic can, transferred from the ocean to the air to form aerosols instead of those productivity of water. I mean, I remember that plot showing the strong correlation, but I was just wondering how that holds for any unproductive water, oligotrophic water, like in Sargasso Sea. Does it? I don't know. I just wonder if anybody look at specifically for that.
Yeah, thanks, Lei. That's a great question. I think Maria can probably tackle that. Oh, you're on mute, Maria.
I don't know if I will cover the full topic, but indeed this is a question, a very good question. It has been also commented during the period that they have shown this nice correlation between the chlorophyll and the organic carbon in the sea salt. And I think that Patricia Quinn and her co-workers worked a lot in the oligotrophic area, and they related the aerosols in the atmosphere mainly to the dissolved organic carbon into the water, and not really the chlorophyll was a better proxy.
Okay.
Let's say. So that's I don't have the paper in mind now to tell you which one, but that's yeah.
Okay.
I believe Jurgita referenced that paper. It was the is chlorophyll a good something? I forget the title's phrase
really?
as a question. Is chlorophyll the right? Yeah, and it's a Patricia Quinn paper. I can add a little bit to that, and then Arna, I'll let you come in. Yeah, so I've also done some work on this at near Batson, Bermuda, which is a very oligotrophic area. And we found chlorophyll was not a very good proxy for organic aerosol concentrations. It required a certain level of primary productivity to be present. So there actually had to be active biological cycling, and that probably gets back to what you're saying about the fact that most of the time it's a fairly oligotrophic area. But people have found relationships with chlorophyll, with DOC concentrations, with POC concentrations, and with primary productivity in varying degrees in varying places. Arna, did you want to add something to that?
Yeah, just the same comment that not just from an aerosol perspective, but also if you want to, tie the PCO2 in the ocean, CO2 sinks in sources, to chlorophyll, which is readily observed, you find that it's rarely a good proxy. So I think chlorophyll, despite being so easily accessible from space, is usually a bad predictor because it, as we've heard, it doesn't. It is a variable that doesn't include the history of dynamics over a couple of weeks or the relevant time scale. And of course, we need other parameters which have more predictive capabilities.
Yeah, it'll be great to integrate those different physical, chemical, biological parameters to see we have probably a big study to see what really important in terms of forming those particles. I have another very general question about the microlayer. So do you know the importance, the relative importance of the microlayer in terms of the enrichment of organic matter? Aerosols, we think about those bubbles, they just transform from the water, then just burst on the surface, right? Does the bubble, those organic matter, they coated during the process, or just the vast majority of the organic matter coated on the microlayer, or is it the whole process, or there's a certain proportion of how important what if there's no microlayer? Sometimes I'm just this is really general question. I'm thinking when the water the bubble just from to the surface of the ocean, it keeps coating organic matter, right? Or it doesn't, or it's only because we know the organic matter is enriched in the microlayer, then the microlayer is the really important factor or player for this coating of organic matter. I'm just.
Thanks. Yeah, it's a good question. Maria or Arna, I'm not sure which of you are better poised to speak to the microlayer. Neither of you.
Not really.
Lisa,
could we?
I mean, it's really a fundamental process, but
Lisa, yes.
Phone a friend from the audience and let Lisa Miller take over here.
Sorry, if it's okay for me to interject. Yeah, the bubbles as they rise do scavenge organic matter from the water, and they can actually be a very important source to organic enrichments in the microlayer. An important thing to keep in mind is the microlayer is always there. Okay, it's just sometimes it's enriched, sometimes it's not. So like, for example, at high wind speeds, you actually find higher microlayer enrichments in organic matter than you do at low wind speeds because of this bubble scavenging process. So you're thinking about it exactly correctly. This is an important phenomenon. So in terms of aerosol production, yeah, the bubbles will burst whatever is already at the surface, but they're also bringing organic matter to the surface themselves. Does that help?
Thanks, Lisa. Excellent answer. Do we have other questions from students? Does anybody have a points of clarity or something that just didn't make sense to them? I know it can be hard to ask those questions sometimes, so I'm actually going to ask one of Maria. That's something that as an atmospheric chemist who studies aerosols, I should know, but I'm going to ask it anyway because this is a safe space. Maria, I'm always confused when people talk about CCN and IMPs as being two very separate things happening in two very separate places, but aren't there species that can be both? And if you can be both, then what controls which one you end up as?
This is an excellent question. I like it. That's, in reality, if you have, you can have an ice nuclei particle. That means one that has it's not soluble most of the time and has the possibility to make ice, but you can age it due to reactions that can happen, and then it can be converted to cloud condensation nuclei, so make warm clouds. So the point between, in reality, it's the same pool of aerosols. And depending on their properties and how they age in the atmosphere, they can act as one or the other, make warm or liquid clouds. And the point is that the ice nuclei are something like 3 to 4 orders of magnitude less abundant in the atmosphere than the cloud condensation nuclei. Can be even more, but that's the order of magnitude, 10,000 less, or yeah, something like that. But you can convert the one to the other. And I have shown a paper, I have discussed a bit this organic coated of ammonium sulfate. Ammonium sulfate is liquid. It's a very nice CCN aerosol. But as soon as you put some organics on it, it can become solid, and then it could probably act, I don't know exactly, but it could. It's to be investigated, could act as ice nuclei. So thanks for the question.
Thank you. Yeah, no, that's excellent. Krista, go ahead.
Hi, everyone. Thank you for your nice talks. I have a question. I don't know if anyone can answer it, but I'm dying to know. So we always hear that the aerosols are the biggest uncertainty with radiative forcing and predicting of global climate change into the future. How do we solve this problem? I mean, what should we do? What is the next thing that we should do to try to answer this uncertainty?
Yeah.
Excellent question. Maria?
Yeah, this is on me, I imagine. So I think this goes two ways. The one is to understand processes better and parameterize better in the models. And that's why we discussed, for instance, the ice nuclei properties of aerosol have to be better represented in the models. The other thing is also to use as much as possible the observations into the models. And we're living in the satellite era, so we should be able to integrate this information into the models and get a better result out of it. So there's a lot of research happening in the aerosol cloud interactions. So understand the processes, how warm clouds are formed, how mixed phase clouds are formed, how secondary ice also is formed, because once you have the first ice crystals, then this can break also and multiply. So I think it's two ways. One is understand processes better, and the other is integrate observations into the models.
So it's basically like every other topic that has the large uncertainty.
Sorry to say that, but that's the way.
No, I'm just teasing. But can I ask a follow-up to the remote sensing question? So how good are the data to be assimilated then from remote sensing? Is this an active, so I don't know, sorry. Is this an active research topic, getting these uncertainties down, or is it satisfactory?
So you want to say data simulation of aerosol products?
I want to say remote sensing of the aerosol properties that are important for assimilation into models.
It is. I mean, see, even in the Copernicus, the European forecasting models, the Copernicus models, they do data simulation of aerosols from in situ, and started doing LiDAR observations also, and also the satellite observations. This is ongoing research also, but it happens.
And the uncertainty is not high, or the uncertainty is?
The uncertainty is very high because the satellite also is a result of a model. Because you have changes in, I mean, what the satellite sees is changes in the radiance. And so you have to convert the optical or whatever signal to concentrations. And this is a model behind it. And another big field of research is to validate the algorithms that convert the satellite observations to something that has a physical meaning for atmospheric chemistry or even for ocean. So there are a lot of validation satellite validation activities with ground-based stations. And in that case, the satellite sees from the top of the atmosphere down, and what the station is measuring is from the surface to the atmosphere. So you try to to converge in the two and develop algorithms that will be able to give the same result in reality
Yeah.
from both ways.
Okay, thank you.
Thanks, Krista. Great question. Lucas, go ahead. Please
Yeah.
introduce yourself if you don't mind.
Hi, I'm Lucas. I'm a master's student at the University of Heidelberg in Germany, and I have a question for Arne. I don't know if we can bring up his presentation again, but on one of your very first slides where you showed figures of chlorophyll and temperature and I believe nutrients on that for that upwelling system on the African coast. And for me, it looked like the chlorophyll and the nutrient concentration was way more intense south of the upwelling system indicated by the temperature. And I was just wondering why that is, or if I missed something during your presentation.
Well, I think it's clearly that the upwelling itself and the productivity are not necessarily co-located, because it needs a time for of course, blooms to develop, sometimes to even need seed populations for plankton growth. So there is a time axis attached to the waters, and the waters don't remain in that place. They typically move out away from the coast, and this can actually be in the form of filaments and rings and eddies. And on the way out, of course, the biology evolves. So I think it's the upwelling itself is where the nutrients are delivered to the system, and then the waters, the upwelled waters, are kind of a part of a complex advection mixing regime, and the productivity itself therefore is spread over huge areas, and that makes it very complicated to even assess the overall effect in terms of biomass production, because it's not staying in the place where the upwelling takes place. It's spread over a huge area, typically in the Atlantic system to the southwest of where the upwelling was, because the surface waters are kind of moving in that direction. Generally moving in that direction, but the eddies can have different trajectories. We have shown eddies, types of eddies, to move even in a west-northwesterly direction. So that's really more complex than that. And since some of these eddies even produce their own little upwelling or downwelling on the way, there is additional components that add to the picture of when and where productivity occurs, and that makes it pretty complex. Clearly, it's not exactly in the space where the upwelling occurs, right?
Yeah, that makes a lot of sense. Thanks a lot.
You're welcome.
Thanks, Lucas. Great question. We have another question from Hashan. Go ahead. We've lost you on video, but I think we'll be able to hear you if you come in.
Yeah.
Oh, there we go.
I have another question to Arne. So apart from the anthropogenic factors that influence the upwelling systems, are there any natural factors that influences the upwelling system? So can we count El Nino events as a fact mechanism that influence the especially Peru currents system? How do you think about it?
I am not an expert in El Nino and the Peruvian upwelling system, but it is very clear that El Nino does influence the activity of the Peruvian upwelling, the Peruvian Chilean upwelling. So that is a pretty clear example. But even in the Atlantic, we have different decadal, subdecadal, multidecadal variability modes, and some of these provide physical settings that influence also the upwelling. So that is actually part of the problem when you analyze wind variability and upwelling intensity variability, to tease out an anthropogenic component and separate it from these longer natural oscillation scales, decadal or even multi-decadal modes, which are present and are known to influence sea surface temperature and things like that in a region, and possibly also in a quantitative sense the upwelling. So right now I think what we're seeing are often combinations of some state of the natural variability, and on top of that, anthropogenic forcing. So I think that's in itself, I think, an important research question to separate the forcings, and very tricky.
Yeah.
Thanks, excellent question. We have time for one more before the break, if anyone has something that they'd like to ask one of our speakers. Questions on topic, methods, strategies. I know it's quite a complex topic. I have one last quick question for Arne, if you could squeeze it in, similar to what Krista asked. So from an upwelling perspective, I personally tend to think of the big impacts, not just on climate, but also on food production and food security. Certainly sitting in South Africa, the Benguela upwelling zone is a really important fishery. And when there are these oxygen depletion events, there are real impacts on real people. You know, this isn't some projected forecast. These are people who need to eat and need to make money for their family. So what do you think are the biggest research questions in upwelling science, sort of as it relates to our ability to make some kind of predictive forecast or something that can really help people understand these oxygen depletion events?
Ooh, that's a big question. I think what we need is a management system for these upwelling system, a management system that's based on evidence and understanding, and not just on, you know, fish catch and stock assessment, but goes more deeply into the driving factors. That's what we need, but that in itself means we need to better understand the systems to be able to predict it. But I think these systems some of them belong to the least well-managed systems, despite the huge catch. There's a lot of things going on, specifically of West Africa, which is not in good shape. So, and since so many people depend on it, this is an important focus area. And I think, and that's something we're kind of trying to design, it may even be a good example of where you need to team up with not just natural scientists, but need economists and social scientists, whom you can sit together to co-design the research questions and come up with projects that not just pay attention to natural science dynamics, but to social dynamics. And that formats that might be able to help is really inclusion of local population to citizen science techniques and things like that, where we not only increase the database, but involve people with a lot of kind of on the ground level, and also make the results more valuable to them. I think that's a pretty vague answer, but I think that's a direction we have to go to get closer to answering the question and getting to the management requirements it needs.
Yeah, I mean, it's as you said before, upwelling is the ultimate cross-cutting theme, and right across from natural science, I think, into the social science. Absolutely. Any other last burning questions before we wrap up and have a break? Well, thanks, everyone. Thank you very much, Arne and Maria, and we'll thank Jurgita in her absence. It's time for us to take a short 10-minute break before the practical session. We will reconvene at 1700 UTC, so whatever that is in your local time, but essentially we'll reconvene on the hour. So please take a break, take care of anything you need, and I look forward to welcoming you back for our practical on science, communication, and the media. Excellent. Welcome back from the break, everyone. Nice to have you back with us. Next up is our fourth practical session on science, communication, and the media. We unfortunately have bad news that Latif Nasser from Radiolab has COVID and is no longer going to be able to join us this evening, this afternoon, this morning, wherever you are. But it is my pleasure to introduce you guys all to Veronique Garçon from CNRS Lagos, and Eric Saltzman from UC Irvine in the US. And Veronique and Eric are going to take you guys through the science, communication, and media practical. I think it's going to be fantastic. I hope you all really enjoy it. And I will see you all at the end. So Vero and Eric, I hand it over to you. Enjoy. I think you'll see the students trickling back from the break. So go ahead and get started when you're ready.
Sure. Thank you, Katie, for that. And sorry that Latif couldn't make it. So maybe we'll tell you how we're going to function for these 2 hours time. So we'll have first a very brief guide towards giving scientific talk in a scientific meetings, which I will do. Then we'll go for a science communication, which is more a guideline towards more diverse audience like media, general public thing. Then you will have a bit of a homework to do on your side. So I think Paul will split the students' attendance in 6 different breakout groups, and you will be working for about 20 minutes, producing a message box. Then you will return with us and we will, give us our feedback on your message box. Actually, the students also, of course, can give their feedbacks to other groups. And after that, we will have Eric who will take over to present some guides and hints to how to write good papers, basically. And it will be a short session with question and answer time. So that's the way we're going to function. I'm at home. My connection is kind of so I hope it will go through, but it was 34 degrees in my lab, so it was far too hot. So I decided to go home. Maybe I'm a biogeochemical oceanographer for training. I'm doing modeling, coupled physical biogeochemical modeling using a variety of tools and observations. Do you see my screen?
Yes, we do.
Okay, great. Sorry about that. I hope it doesn't happen again. So full screen, is that okay?
But it's not in presentation. Okay, now, yes.
Okay. So we're going to give you a few hints on how to prepare and make presentation. So this very first slide, I think, really set the scene. Today we learn about presentation, and you have a guy first row is saying it's better be interesting because my time is valuable. And so it's very important when you give a talk, get prepared because you're taking people's time. And you should also have good connection, actually. But so that's very important. You have to be prepared because time is the most precious things people have nowadays, and they better learn something. So that's very important. Time is precious. So today I'm going to give you a few basics for oral presentation, on the format and on the science. Okay, it doesn't work. Shit. Oh, okay. So about format, a few words about physical motion and general behavior and technical details. And then about the science. So I want to really say here that it's always stressful to give an oral presentation, in particular where it's not in your native language. So there is no way you're going to get rid of your stress. It'll be there. It can be that you need to go 20 times to the restroom before giving your talk, or you start sweating like crazy. There are different signs of stress. It's very personal dependent, but it's there. So the way to try to overcome that is try to turn that nervous energy into a constructive enthusiasm. If you are here, it's because maybe you want to become a scientist, and we are here about communicating. So you communicate through articles, talks in conference or via Zooms, and through posters. And the only way to get rid of, you will not get rid of stress, but to manage it is to practice again and again. So at least you know your talk, you feel secure about your talk, and then you can concentrate on the questions. But remember, you will never get rid of stress. It's like a performer in a theater is always a bit anxious when he's entering the stage. So I'm trying about the format. I'm trying to get rid of that. So you need to be convincing, dynamic, and enthusiastic. If you don't look convinced when you give your talk, there is no way you're going to convince your audience. You need to be a bit mobile on stage, and remember, you're giving a performance. So I know maybe many of you haven't been in a meeting for 2 years because of COVID, and you will be very happy to be back to conference. And I'm sure you will look very dynamic and enthusiastic. The second thing is, if it works, well, it seems that my screen doesn't want to go. Look at people. It's very important to look at people. Don't put hand into your pockets. Never turn your back to the audience. These are very basics one. In order to not put your hand into the pocket, usually you use a leather or a stick that keeps at least one of your hand busy, and maybe in the other one you will have the microphone, so you're safe on that side. You need to articulate, speak clear and loud, so don't mumble, because the non-native will not understand, and even the native, so it's very important to articulate clearly. You should also get intonation into your voice, no monotonous speech. So the best way is try to kind of build question answers. So people, when they hear a question, your voice is going and they are waiting for an answer. Remember, you're telling a story, so you need to catch the audience attention at the very first slide and keep it alive until the very last slide. It's like, I don't know if some of you have kids, when you go to bed, you tell a story and at least you want to have him listen by the end of the story. So be self-confident. You have been working, this is your work for months and years, so you know what you have been doing. So be self-confident, but I would advise to always remain humble. Humility is a very important quality. Never be arrogant. That will not help you anywhere. And the most important is, as a scientist, be ready and open to critics, constructive critics, of course. The other ones, the negative, don't get upset. Do not talk anything personally. You should detach for your work. I mean, people, when they give critics, it's to your work, it's not to your own person. So I know sometimes it's difficult because you put a lot of effort into and then suddenly when you have somebody saying, wow, this is not, you should have done that, you should have done the other way. And you may feel upset, but I think being a scientist is being able to receive critics, and sometimes you will get compliments. So there is still hope. The last thing is you should learn to respect time constraint, which is imposed by the exercise. If you're given a 15 minutes talk, you don't go 17, you don't go 13, you go 15. It's a matter of respect of the audience, and this is the exercise which has been given to you. So try to learn in a couple, plus or minus 10 seconds, but the only way to be able to respect time constraint is at least practice. So sometimes you may have questions and you have no clue how to answer, right? So if you are in a small lab meeting, you can say, oh, I'm sorry, I don't know what to answer to your question. I don't know, I haven't tried, I don't know. Maybe in bigger meeting, you can try to turn that into a response like, oh, this is a very good point you mentioned. This is something we should really further investigate. That's very interesting. Thanks a lot. The person who asks you the question feels really happy because you made a good comment, and you don't look like, oh, sorry, I don't know the answer. So it's a kind of trade-off, right? So technically, don't overcrowd your slide. It's just one message per slide. I hope. Don't write and then read long sentences. So sometimes people, they feel insecure, so they put a lot of text on their slide with the small figures, and then you lose your audience if you do so. So favor bullet point message and take a message in a slide where you have mostly figures or scheme or whatever. Important also, you need to have consistency between what you say. If you show something and your talk is about something else, then people are trying to figure out what the connection between the two and they don't find it, and then you lose them. So remember, storytelling, you catch the people first slide, you keep them to the very last slide. A storyteller, of course, there should be a logical path in your talk. So you need those transitions, those links in between slides so that the sequence looks very natural. Of course, large character, easy reading policy. People like us, the old one, we wear glasses, we don't see very well. So it's good when the letters are really big. Use appropriate color codes, label access, put units so people, they know what they look at. And most importantly, when you present result figures, don't start right away describing the result. You need first to tell the people what they're looking at, and then you go into the science that it's inside the figure. So people prefer dark background and clear letters. You can like clear backgrounds and dark letters. This is just a matter of taste, but you need contrast so that the message is really visible. Importantly, in French, we say le fond et la forme, which is content and visual. So some people have very nice, very sophisticated PowerPoint, but the science in it is kind of poor. So I would say to make a good story, you need a good visual and a good content. And one should not compensate for the other. It's important to get both. And this is what I'm saying here. What matters really is a scientific, innovative result on your talk. So about the science. So another presentation is like a short article if you have 10 minutes or 15 minutes. So first slide, who you are, where you work, who are your collaborators. Do not forget, you have advisor, you have people who have been helping you. So it's always nice to put all your co-authors. If you give a small talk, you don't need to come up with a logical outline of your talk. It's when you give a seminar 45 minutes, then it's good to have a logical outline of your talk. So people can really walk with you through the talk. So then why you do that? What is the scientific rationale? Either an existing controversy, a mystery to be unrevealed that you need to contribute to, and eventually some specific questions. So avoid coming up with like 10 catalog style questions, because in 15 minutes, there is no way you can answer those 10 questions, and your conclusion will look totally different with your initial slide on the scientific rationale. So there should be consistency between your first slide where you put your question and the conclusion. So, and it's good if there is a controversy, because it's like about the storytelling. It's exciting. It's not exciting, but it raises curiosity of people, because you know there is some people find that, some other find that, and we're going to show that we found what the solution is. So of course you say where you work. I'm working in the Arabian Sea, or I've been looking at future scenario under STP 8.5. I'm giving the methods, I'm giving the models and the data source, but that should be really short. In a talk like that, what matters is really explain why you do that, what is the controversy, the method should be really short. And what matters is really the result and the discussion. So that where most of the time should be spent on. So the selection of key result, you cannot put all the figures you have in your paper into a 10-minute talk. So you really need to make the selection of what is really key, important, present as clearly as possible. You can discuss at the same time or add a couple of slides for discussing some of the issues. And you conclude. So of course you may acknowledge your supporting agency. It's always good to thank the people who give you the money to do the work. And your conclusion should really mirror the introduction and the question you had at the beginning. So people, they are at the start of the story, they see the end, even if you not answer all of it, but at least they see where you are into the answering your initial questions. I'll close up here. So I will thank you for attention. So usually what we do in the real SOLAS summer school in presential, then all the students of the group, they go on stage and they make a short presentation and we give them, feedback on the spot. So my understanding is that later today, you will get an opportunity to practice during your flash presentation for your poster. So that would be a good way to practice. And I will close that presentation here if you have any question, and then I will present the one on science communication. So this one is really devoted towards talks in scientific meetings, workshop, conference, symposium. The other one is more aiming towards different type of audiences. So doing that. And so I do not see all the answers and discussion. Okay, no questions so far. Okay, it doesn't really appeal for question anyway. Okay, so let's go to the other PowerPoint. I hope I don't have problem sharing. Do you see it? No.
Yeah, I think it's just slow.
Yeah, it is very slow. So do you see it now?
Yes,
Yes.
not in presentation.
Okay, so I'm putting in presentation mode. Is that okay? Okay, okay. So now it's more gearing towards communicating science to different type of audience. So it could be media, journalists, TV, journalists from a weekly magazine or daily newspaper or cultural magazine. And it can be policymakers, it can be the general public, it can be high school teachers, it can be kids in high school. So very different audience. And basically is how to explain the science you do, for instance, to your parents in very simple words, which is not very easy, I agree. So it happened to me the first, usually I honestly, I try to avoid media because I just don't know how to handle it. So the first time I had to do it was after our science article in January 18, where we published on the loss of oxygen in the ocean. There was a lot of authors, but I was the only French author. So finally, I got all the French newspaper, that was a weekly magazine from Brittany, and they were really interested because of green algae in Brittany and the anoxia over there. We had newspapers, so a brand, and I had to face it. So I had to explain in very simple words what we were doing. I had also to write an article with Vladimir Yabinin, who is the executive secretary of the IUC UNESCO. So stress, I would say. And I realized it's a whole different job. And what I learned is when journalists, so you receive those emails or those phone calls, never answer on the spot. You take an appointment for the next day. So at least you have time to think it over and try to make your wording a bit easier without jargon, with a very simple word. So never answer right away. and always ask for rereading the article. Sometimes now, I must say, at least the French journalists are really doing that. But I know some of my colleagues, sometimes they were discovering very strange things they never said in the article. So it's always better to ask for rereading the article before it goes out. So that the other two too. So I'm just putting a couple of things. So year 18 was a bit difficult for me. But why? Because basically you're escaping from your ivory tower, your very cozy office or your very cozy home office where you do your research, and you don't have to matter and to worry about making your science matter. You know, it matters for you, you know, it matters for your institution, but why should I worry about why it would matter to people? So yeah, it's a bit of an adventure. So I was one of the co-governor of the effect of climate change on the world ocean symposium, which took place in Washington, DC in 2018. The next one is in Norway next year. And actually the US-based company Compass, who is about communication partnership for science and the sea, gave us a very nice training about leadership and communication. And I thought that was very useful. So this is what I'm going to try to present to you now. And hopefully after that, you will be better than before. So I think the whole story is really here on that slide. Basically, the first presentation I made to you was on green pyramid, the scientist pyramid, right? You start describing the background, the rational, controversy, then you go for your supporting details, your methods, what you've been doing in your work, then you present your result, and then you conclude. This does not work with different audience than scientists. So you should think about inverting your pyramid and thinking and looking at the red pyramid, where you really start with the bottom line. Then you do the so what. So why the audience should care about you tell them. And then ultimately you can go for the supporting details. So the so what is very important. And the so what is really directed toward the audience you are targeting. You won't have the same so what for kids in the school than policymakers or journalists that would communicate with a broad audience. So the Compass company, they came up with that message box, which in a way is a translation tool between what's on the left side here, the classical scientific title, abstract, background, method, result, discussion, conclusion, and then according with a different audience, you need to transfer that into that message box. So the center is really what is the big issue? What are the relevant problems associated to that issue? Then the so what, why people should matter of what you tell them. What are the possible solutions that you could implement, they could help you implement? What are the benefits they can draw from those solutions, basically? So the message box is a very simple tool for communicating complex ideas. It really will help you to organize your thought and identify the key points. It helps you to be flexible because you can use it to structure a presentation, outline a proposal, or prepare for an interview. And remember, the audience can only absorb a limited amount of information. You have been working on a topic for months and years, so you know all the tiny details, but people in 10, 20, 15 minutes, they need to absorb all of it. So it's very difficult. So you need to really streamline what really matters to them and what do they need to know, especially. So you need to distill your information into concise messages by answering the following question. What is the issue and problem? Is there a conflict? Is there a controversy? What decision should be made? Then the so what? Why does this matter to my listener? Why they should worry about that? And then the solution, what action do I want my listener to take or support me to do? And what are the benefits? So what would my listener benefit by resolving the problem? And remember, always keep your audience needs in mind. And they are very different according to each audience. So you need to pare down your ideas so that each of these 4 questions can be answers into 1 or 2 concise sentences. If you end up with a paragraph, you need to keep working. Let's see, I think there is. You need to keep working to refine your messages, and you keep practicing your delivery. So you tell your story to your mom and dad or boyfriend. If they get the message, that's fine. If they still don't get it, you need to keep refining it, and they will evolve and get better over time. Doesn't want to go. I can't get my next slide to come. I don't know why. Next slide is not coming. Okay, it is. So this is an example of one message box where the issue is hypoxia. So there's a lack of oxygen in water, which is increasing in the coastal zone of the Black Sea. And the work has identified over 100 sites that have hypoxia, and the occurrence is steadily increasing with time. So the problem related to that is that dead zones are now spreading in coastal areas of the Baltic Sea, whether it was a problem in the open Baltic Sea, but now the coast is suffering from the lack of oxygen, and this is new. So what? 2 things. The lack of oxygen kills fish in coastal area, and the coastal zone are important nursery area for small fish, especially cod. So in addition, coastal hypoxia means more algal blooms, which means poor condition for swimming, affecting tourism. So what are the possible solutions? So the politicians need to act to reduce nutrients. And the Baltic Sea Action Plan has identified how much needs to be reduced, but the country need to implement the plan now to reduce the nutrient loads. So you need to address the large animal facility, the sewage treatment plant, especially along the eastern shore of the Baltic Sea. And the benefits will be having oxygen back in the coastal zone, would be in a better functioning ecosystem with less algal blooms. So this is a very dense message block, far too dense to be understood by an audience. So to work on your message, you need to simplify it, maybe get it onto the paper, avoid any kind of jargon, because here the audience was the general public. And as I said, you try it out to your relative. Are you understood or not? If not, you rewrite and you try it out again until you're happy with that. You need to think of analogy and metaphors, because real life examples are really great when you talk to the public. And think of very catchy phrases, like a good pitch. That's very important when you talk to journalists. Of course, you can try to do it without the message box, but the issue, problems, so what, solution, and benefits are really the natural streamline for building such a message box. So this is a streamlined message box, which kind of fit what we would say would be a nice message box for an audience like the public. So the issue is coastal hypoxia is common in the Baltic, and that was not previously known. The relevant problem is dead zone are spreading in all the coastal zone of the Baltic Sea. The so what? So fish kills because of lack of oxygen, and more algae blooms that are produced by coastal hypoxia. Solution, politicians need to act to reduce nutrients now. And the benefits, it would be having oxygen in the coastal zone would mean a better functioning system with more fish and less algae blooms. So what we got to do now, it's going to be your turn. So Paul will try to make you in 6 different breakout groups. And you can prepare a message box. So remember the topic, it's problem, issue, so what, solution, and benefits. You can pick up a topic you want to communicate on. A SOLAS topic, a SOLAS sounds topic would be nice. And we leave you about 20 minutes to write such a message box. We think of the audience to target being journalists today. And then after 20 minutes, we'll be reconvening, and each group will present its pitch. So it would be like 5 minutes for each group. It's more or less 5 minutes. I think we're good in time. And so, yeah, we may have more than 5 minutes for each group. So I don't know how many you will be per group, but if each one of you want to give one part of the message box or just one person presenting the whole message box, it's really up to you. So I'm leaving now the floor to Paul, who might want to explain how to proceed now.
Hi there, Vero. I have moved everyone into the breakout rooms. They're fairly well practiced at this now because we've done it for all the other practicals. So I don't think they would need much instruction, so I've moved them there. Just to note that I've only made 4 rooms instead of 6 because there were fewer students than expected. So they're in groups of sort of about 5.
Okay, so 4 rooms. So we'll have more time, which is good.
Yeah, always good.
Paul,
hi there.
question. Is there, did you put a strict time on this so their groups will end at 20?
Yeah,
No,
we
no,
don't have a time. Yeah.
So how does it end?
We can decide. So it can be totally flexible. Would you like, at the minute, I will shut it at, well, in 20 minutes, which would be at 7, well, on the hour.
Maybe 20 minutes, maybe we can give them 25 minutes until 8:05.
Is there a warning thing where you can say closing in 5 or 4?
Yeah, I'll do all of that. If you let me know when you'd like it to close, and we'll do 10-minute warnings, 5 minutes, and then they're given a few bits at the end. But we can be totally flexible to whatever you'd like.
Okay, so maybe what I'll do is put back the message box so that they have the the issue problems. So they have it doesn't need to be with that. We just need the different section. Okay.
Yeah, for sure.
Okay, so see you in 25 minutes, more or less. Okay.
Yeah, no problem at all. So
but wait, sorry. Is anyone going to pop into the rooms to check out that the students are talking?
Yeah, I think Vero should go pop in.
Okay, everyone is back. Do you hear me? Okay, so maybe we'll start by listening to group 1 to present their message box. I don't know if you have it like with a template or you just want to set it orally. So who's going to talk in group 1? So who is volunteer or who decided to talk?
I don't even know who is group 1.
No, we don't know what group we were either.
Okay, so group 1 is Anna, Lunde,
that's me.
Sam Terimozu, Ashan Lei, and Thais Lobato Sarmanto. Sorry for the pronunciation. So who is presenting? Anna?
I can present since I'm talking. If no one else wants to present.
I don't know. What do
Lei, do you want to present?
No, you can go ahead. I want to do that.
Okay, so everyone in my group, feel free to intervene.
Sure.
Let's see. I can share my screen since we,
Sure. Sure.
So I'm not going to put it up on full screen because then I can't see anyone, but I hope it's okay.
Sure, go ahead.
Yes, so we had a very diverse group with oceanographer, meteorologist, environmental scientist, chemist. So we struggled a bit maybe to find like a common grounds except for this SOLAS community, but then we said that our issue is ocean acidification. The problems with that would be dissolution of corals and other calcifiers, of course. So what? Then we have the reduced shellfish and fish stocks and the degradation of ecosystems. So some solutions would be to reduce or stop carbon dioxide emissions and support innovations that would stop these emissions. And the benefits from this would be sustained livelihood for those who are dependent of oceans, which is most people, and then also maintain resilience of globally. Yes, thank you.
Yeah, no, I think that's a very good one. I was just curious, what are you thinking about stop innovation, support innovation that stop carbon emission? What do you think of?
I mean, we didn't think of a specific. We discussed a lot of different options, but just to reduce or stop carbon. If we talk to a politician and want to say something more concrete, then maybe it would be better to have not only to stop something, but to support better things, basically, not to be so negative and say that we have to stop everything. Also that we should support like other technologies. We didn't have time to discuss in detail what those were, but yeah.
So remember, the audience were journalists, so that would be the general public. And I think that last sentence was people would ask what you mean. So you simpler words, really describing what you intend to put when you do support innovation to stop carbon emissions. But the other ones are really simple, clear to the point, I think. If Eric, you want to add anything, or feel free.
Or anyone in my group.
Or anyone in your group, of course. But for me, it was good. Just the last part, the last bit for solution, which was not explicit enough for general audience, right, by journalists. Okay. So maybe we'll go to the next group. Thank you so much. That was great. That was a great one. So group 2 is Alicia, Guadalupe Uribe, Lopez, Lou Coronado, and Filipa Ricard. So I don't know who is presenting.
Hi, everyone. Filipa and I are gonna cover the theme because Lou is facing some connection problems.
Okay.
So the problem the issue we covered was that it can be expensive to make accurate measurements on variables such as PCO2, for example. And the problem is that most sensors are quite expensive and sometimes they break. So it can be difficult to repair them because of logistical, financial, or other reasons. And the so what is that, we always need more data, and cruises are sometimes complicated, and coastal areas, for example, are not entirely covered. And Filipa is gonna continue with it.
Yeah, so the solutions we were chatting about was that commonly available sensors on, say, eBay or Amazon can actually be made good enough when we build accurate methodologies around their capabilities. And the benefits of this are that you can make scientifically robust and affordable measurements, but you can also be a powerful tool for community engagement, which empowers local communities to be part of the science. And that links back to not what we would say to journalists, but it links back to the sessions yesterday where we were talking about the social ethics around actually going out somewhere and taking samples. If we can empower the local community to take those samples and provide the data and be involved in the science, then it makes science better all around. And if it's an affordable sensor, they don't need specialist intervention to fix. And that's it.
Okay, I think that's great. That was a lot of words. So you would need to work on putting that in very few words into a message box. And I think I'm really interested in that one because, like I was telling you, I'm part of ORS on oceanification, and we are about exactly the same problem you were tackling, how to increase observing capability in developing country when we have equipment which is very expensive, and when you have instruments that are breaking, they need to be sent. And so I would be delighted to have a message box on that, so you can send it by email. That would be great. So it's great, but you need to phrase it in nice words. So pick up the right words, but the whole story is really excellent, I think. So that was very interesting.
Okay, great. Thanks, Viva.
Thank you.
So I'm waiting for your message box. So we go to group 3. So group 3 is, sorry for pronunciation, Arayeta, Emmanuel Chevasus, Juan Camilo Torres Lasso, and Raiza Hill.
Yeah, so I'll be presenting because Arayeta it's having some audio problems as well.
Okay, I know that.
So can you see the message box?
Yeah.
So it was ocean acidification too. So it's an issue because it's the increasing pH of ocean waters due to the high concentrations of CO2 in the atmosphere. The problem is that these affect the whole ocean marine life and also reduces the ocean's power as a CO2 sink. And so what is that the ocean is one of the main sinks of CO2 in our biosphere, and that would completely improve or at least not sustain the climate change. And also, since it will affect marine life, it would alter him through the supply. So for the solutions, we thought about the main ones that would be reducing the CO2 emissions and also the monitoring, because we can't manage what we don't measure. So we need to provide funding, especially in places where the communities are more impacted with the high pH waters. The benefits would be to make better decisions and better prognostics of what would happen with this increasing pH. And it will also reduce the extinction of species and the complications of that. So
you meaning decrease pH, not increase pH?
Yeah. Yes, sorry. Okay. Yes, exactly.
Okay. That's important to get the issue right at the beginning.
Yeah, sorry.
Okay. No problem. The benefit, better management, reduce extension of species, better management of the impact. What do you really mean? Remember, this is targeted towards journalists that will go to the general public. So I think all the rest is easy to understand. Monitoring, maybe you need to say exactly. So it would be monitoring pH in ocean water. Is that what you mean?
Yeah.
Okay. And a better management of the impact. What do you exactly mean by that?
Would be like, since it would impact marine communities that are sometimes used as human food supply, it would be to have to manage that and how you be able to compensate these communities mainly.
Yeah, I'm not sure I really understand, because you will have species that will be impacted by a decreased pH that might suffer from that. But yeah, I'm a bit puzzled by this one. Reduce extension of species. Yeah, you might have species that are most affected than other. I mean, with OA, you have always winners and losers amongst marine species. Some are affected, some are less, and some are even more favorable, like jellyfish, for instance. They're really doing well when everything is going bad. So I would, if I were you, I would try to rephrase, because better management of impact is really very general. It might talk to the people. So be more with more real world. What you exactly mean? That's I mean, the rest is and monitoring pH. So you need to know to say what you will monitor. Marine life, I think, yeah, that's good.
Okay.
I accept that one. Okay. So I don't know. Are we done with all the? Yeah, we're done with all the groups. So so we don't have time, but what we were doing in the last summer school, we organized. We were giving people would take the same topic, but with a different target audience. Like, for instance, policymakers. So how would you change your wording in order to tackle a different audience? So I guess the take-home message of that science communication is, when you talk to press and media, never answer on the spot. Take time, think it over, take an appointment for the next day. I mean, if it's a hot topic, journalists won't wait for a week, right? So, but the next day, so you have time to think of what you want to do. And when you deliver your message, think of the audience you want to give and why that should matter to the audience you're aiming to address. I've seen a hand by, I've seen Christine.
It was me. Does group 4 not want to present?
We have 4 groups, right?
Yeah, did group 4 want to present or not? We didn't get the last group.
Sorry, group 4. I thought we did all the group. Sorry, thanks, Krista. So group 4 is Axel, Hao, Josefa, Lucas, and Niels. Sorry, guys. Go ahead. So who is presenting? Niels, Lucas?
I think we didn't discuss who's presenting, but I can go ahead.
Okay, go,
share the
Lucas.
screen. We didn't really have time to like go over it again, and we took a very general topic. So our issue was that the atmospheric CH4 concentration significantly increased in the past years. And of course, the problems associated with that is that CH4 is a potent greenhouse gas, which further drives climate change and intensifies related issues like weather extremes, temperature rise, floods, sea level rise, and so on. And why the society or the journalists should worry about that is that it is a threat to ecosystems and food and water security and social stability. The solutions that we came up with are green energy, of course, which if we don't have that many coal power plants, for example, anymore, then we wouldn't emit that much CH4. Geoengineering, we could reduce kettles, or we could make changes in what we feed the kettle with, and also nature-based solutions, like, for example, reforestation, restoration of wetlands, or restoration of kelps. And the benefits of changing that would be, of course, the slowdown in global warming and the protection of our living environment and our society.
Okay, so maybe what I would just go straight to intensify issue. Remember, you need few words. So maybe you could say directly it intensify related issue like weather extreme, temperature rise, floods, sea level rise, etc.
Sorry, just noticing.
You have a list of solutions. So would you be, it's a long list. Of course, they're all good, but would you put a hierarchy, or maybe because geoengineering, are you sure that would talk to the general public?
I didn't know who put that down, so maybe someone wants to comment on that in my group.
Yeah, I think I will go rather directly to reduce or changes in cattle diet, for example,
Yeah.
and nature-based solution. I think this, or restoration of wetlands. Probably these two, I think it will bring more attention to the audience or people who are attending, or listening to.
Yeah, fully agree. Also, the other ones are important, but here, if you really want to touch the general audience, is reduce cattle, changing cattle diet. That speaks to everybody now. And a nature-based solution, I would go, because geoengineering, people, I'm not sure what it is. It's kind of our jargon words. Green, I think everybody knows, but I would go for those reduce cattle and nature-based solution. That would be more easier to target. Okay. Okay, great. So finally, you choose methane over plastic. Okay, sorry, I thought we went through the free groups. I forgot you guys, but I think you did. We had quite a variety of topics, so I think that was good. Great. So maybe now I leave the floor to Eric. We're just in time.
Okay, perfect. That was great. Krista, did we want to do a break? Give them 5 minutes?
I think I would say yes. A 5-minute break is
okay. Everybody always appreciates a break. So take 5 minutes and come back. It's 11:24. So come back at 11:29. Okay, are we back?
Yes.
Is everybody back? Turn on your cameras. We're a nice small group. I want to see everybody I can see. If you have bandwidth problems, I understand. If you're in your pajamas, we don't care. We don't mind. Excellent. Keep coming. Keep coming. We're almost here. Okay, so we're a smaller group than I feared, so that's awesome and really good. So let me try and share my screen and tell you what we're doing here. Okay, share. Okay. Can you all see that?
Yes.
You can shout. It's good if you shout. Okay. Hold on a second. I want to see. Oops. Hang on, I got to get my view in order here so I can see everybody at the same time. I'm looking at my thing. Okay. Okay. There we go. All right. So this, if I resize this, does it get smaller for you guys? It is getting smaller. Okay. Too small? Good.
That's better.
I am not seeing it the way you were seeing it. So for whatever reason. Okay. So this is a little bit practical about writing, but here we're focused on academic writing. Okay. So the kind of writing you got to do in your career to get done what you need to accomplish to get a degree, to get a job, and that kind of stuff. And writing like an academic, like a scientist, right? We want to be professional writing, but professional scientist type writing. Okay, so before I start, I want to ask you guys who absolutely loves writing in their career? Raise your hands. One. Not everybody. I'm not seeing. Maybe some cheers. We love writing. Okay, who hates writing? Oh, Josefa, you're on the low side. What? One hater? Only one hater? Two haters. Come on, there's got to be more haters than two in one of these things. Okay, so I've been teaching these practicals at SOLAS summer schools for a long time. I've met a lot of students. It's always really interesting to hear what they think about writing and what their struggles are and so forth. And I also teach writing back at my home institution. So I'm constantly learning from them what their struggles are. And so it's really important for me to understand what you guys think about when you're trying to write. Okay, so I wanted to do a quick breakout session for you guys to just get down on paper what your biggest challenges are. Okay, what are your daily struggles? What's pushing back against you? What prevents you from raising your hand and saying, I love writing, when I ask that question? Okay, so don't filter yourself, just get it down. Have one person in the group be a note taker. And then when the breakout ends, because it's going to be fast, when the breakout ends, paste your notes in the chat so we capture a record of that, because I collect these things. I have them for years, and then we can look at them briefly. Okay, so let's do that real quick. Paul, are you there? Whoa, he's quick.
Okay. Go on, it's happening. Yes. I will also save all of the chat and send that to you afterwards. So don't worry about copying and pasting. Anything that goes in the chat, I will save and I'll send it all out to you afterwards.
Okay. In general, I think 5 is a great number for these breakouts. I see you have 6 in a couple, but 5. Perfect.
Perfect. And then do you want different groups every time?
Yeah.
We'll mix them all up around. Brilliant.
Yeah.
No problem. Okay. And how long would you like them in this breakout for?
5. Just
5 minutes. Yeah. No problem.
Pull the plug at 5.
Cool. I'll drag them back.
They have to go quicker later in later breakouts, but we'll start with 5.
No problem at all.
Wish I could make my screen show the same thing that.
So Eric, you did something and it made your what you're showing us bigger. So I don't know what the last thing you did was.
Bigger meaning better or worse?
Better. More. It's covering more of the screen. It looks so. Right now, it's very narrow in the screen.
If you'd prefer, I could share your slides on my screen, but you could still control them. That would be better.
I would totally like that, but I want you to use the same file that I have.
Is it the one that you emailed me
No,
yesterday?
it's slightly different. It's slightly different.
This is a real challenge for us. You want us to plan ahead, and we can't.
Hold on. Well, maybe we can get it done in real time.
No problem.
Oh, can I paste it in the chat?
You might be able to upload it. There's a little a button that looks like a document with a folded corner. And if you click that, you should be able to upload a document to the chat.
Wow, that didn't work. That didn't work at all. I'm going to email it to you.
Okay, I'll get it up. Becky, could you get a Google Drive link as a backup in case the email doesn't work, please?
Oh, maybe it worked. Did you just get a file from me?
Got it. Yes.
Okay, I'm sending you to PPTX as well, but just use the PDF. It's fine.
Okay. Let me get that up.
I am going to stop sharing, or did you stop sharing me?
You stopped.
Unbelievable. It's like every day is a new day. Not like I haven't done this a million times already.
Okay. So just to test it, if I give you slide control, there you are. So you should be able to control my screen now.
Wow.
Oops. Maybe it's too little. Yeah. Perfect.
Yeah, it works.
There you go. And that has been about 5 minutes. So I will bring everyone back into the room and give you control.
Yeah. Okay. I hope that exit wasn't too jarring for everybody. I know that can be kind of like abrupt when you get vanished out of your group. Okay, so I would just love to hear from each group and lay it on us. Group one. Do you know what groups you were? Maybe not. Paul, help us.
I could normally help you. Unfortunately, I've started randomizing the groups ready for the next section. So I have forgotten as well.
Oh,
and
wow.
What we normally do.
That's okay.
What we do in this situation, normally what we advise in our professional opinion is to pick on people who have got their cameras on. So I would hand that over
That's
to you.
incentive to turn off your cameras.
I think we
in
are
which case, pick on
group
people with them
one.
off.
Okay, let's just go in real time. I want to hear what you guys talked about in your groups. Go ahead. Take it.
Do you want us to put it in the chat or just chat?
Yeah, just go ahead. Tell us.
Okay, so the first one we talked about was like actually having time to write, and that isn't time management. That is like the literal time set aside to write. We find quite challenging.
Because you were doing other things, you're asked to do other things?
Yeah. I guess. And another one that the group were talking about was anxiety around citations, that like whether you've got all of the relevant ones in or you've missed a specific one, or what was it? Hang on, I've got notes.
That's awesome. That's a first. citation anxiety. I have never heard that one in all these years. That's cool.
Yeah, and then justifying method development with citations. And I guess like that's what we do in science. We develop methods, right? But sometimes there's more anxiety around that because it opens you up for criticism.
Oh, interesting.
When you're just a little PhD student or something, then it's scary.
Okay, so just so you know, little PhD students are the people who actually do the science in this world. So don't downplay yourself, right? If it wasn't for you guys, nothing would get done. Okay, somebody else.
That's why we have no time, right?
Huh?
That's why we have no time to write.
Yeah, exactly. Okay, somebody else from the same group or different group, doesn't matter.
So, hey, everybody, so to begin with the writing, right? You have collected your data, then you make plots, right? So you build a story around these plots. So what I have been struggling with is how to keep everything, concise. How to talk about your results, as short as possible.
Yep.
Sometimes your draft is just, there's some information that is just redundant. I mean, something you can really just keep your manuscript as short as you wish, but some just how to word, how to phrase, how to discuss.
Okay, Lee, you're describing a really common problem in writing.
That's the first struggle.
My paper is out of control. Like my paper's like going on forever and I can't get a grip. And how do I get this thing under control? That's really common. Okay, let's keep going. Collect lists.
There's another one. There's not a struggle. So sometimes you just want to do as much and talk about as much as you can with your data, right? But for some fundamental data sets, there's only that much you can say. I mean, how you bring your data or to the next level, if that's possible. You know what I mean? So you want to talk too big of your research results. I mean, I'm not sure if I'm presenting that clearly.
Oh, that's okay. I think you're describing one or two end members. There's I just have too much to say. I can't say everything in this paper. And then there's the other end is that I have nothing to say. What am I going to put in this paper? There's nothing. So that's great. I definitely have heard that from a lot of students. Yep.
Yeah, that's my notes.
I hope you've written these down and we'll paste them into the chat at some point. It could be a little bit later. That's fine. Josefa, you're looking like
I was thinking that a common problem could be also be that the text is never good enough to show it to your co-author or supervisors or other people. Like you never think.
Anxiety.
Or advisor anxiety.
Okay.
The text is never good enough. It could also be better, and then you keep it to yourself, and you are afraid to show it.
Yes, that's deadly. Right. Yes. Okay. Keep going. Next.
I can add some things that came up also in our group. One that was new to me that someone said was, I think you called it gatekeeping. Like the same formulations and jargon is always expected, and it feels like you have to live up to using certain standard phrases. Or you who said it, feel free to fill in if I misunderstood you.
That was me actually. So what I want to say about it, while writing scientific papers, often the language doesn't seem very natural. It makes it hard for most people that are not well versed into the subject to read. There are certain formulas that we always use. If you work on a certain subject, you will always see the same introduction, basically the same thing. It's always the same thing that is expected, which can get really, really, like, you first have to learn how to write exactly how those papers want you to write before starting to write, which can get really annoying.
Interesting. Writing how the papers want you to write. That's a first.
That's what I experienced before, so that came to mind.
Interesting, but definitely things are very formulaic in certain fields, and you see the same language again and again. That's absolutely true. Let's keep going. Others, let's take a couple more.
I usually find it very hard to focus in my head, but also focus the text. We talked about that also in my group, like getting all the pieces together and find a good narrative can create some, I wouldn't call it anxiety, but it's just the issue of focusing is for me a big thing.
Yeah, let's call
you
that
put a lot of
finding your narrative.
Yeah.
Right. And how?
And then you change that 1,000 times before you're ready. It feels like as well. So not 1,000, but many.
Okay, let's jump to the presentation, Paul. Oh, look at that. So this is amazing. He's showing the presentation, but I have control of the pages. Okay, so I just wanted to show you some that I collected from earlier workshops. Not by no means complete list, but just some. One would be, it's taking too long. My manuscript's out of control. I don't know where it's going. I used to be a good writer. I thought I was a good writer, but now I stink. Why so many drafts? My advisor, wants me to write 50 drafts of this paper. How do I deal with my co-authors? My friend wrote his paper in 2 weeks. It's taking me months. My advisor, he hates what I write, but all he'll do is rewrite it. He won't actually teach me how to write. And there was one great discussion about, I don't believe what I'm writing. Like, my co-authors believe it, but I'm dubious. I don't actually believe the science. So we could go on for pages. There's lots of issues in writing. They're all interesting and they're all common. All we as scientists have lots of these issues. We're not going to solve them all today, sadly. Okay, I want to jump, but what I am going to do later is talk to you about my sort of technique for overcoming a lot of these issues, okay? But before we do that, I want to break out on a completely different subject that I think interests young scientists quite a bit. So I want to kick you out for 5 more minutes for a conversation on this subject, quite different. That is, what's the criteria for co-authorship on a paper? So as young scientists, you're going to find yourself invited to co-author or people asking you to co-author. And you're also going to have your own manuscript and be trying to figure out who should go on this authorship list. And so how do you think about that? Okay. 5 minutes. Ooh, maybe we should do 3 minutes because time's got to move marching on here. 3 minutes.
I've sent people off, so that is fine. They're out and about in their breakouts. Just as a note that we have fewer posters than we were expecting for after this practical session. So, and with the break at 1/2 the hour, I think we can skip that break and run this practical overtime. So don't worry too much about finishing in 7 minutes. I think that the students are engaging well with this. So take the extra time to do it properly, if that's okay with you, Krista.
That is hugely okay with me.
Perfect. Oh, sorry, hay fever is killing me, but we will grab people back in a couple of minutes' time.
Yep. They're funny.
This is great, Eric. I'm taking notes. I never made it into your sessions I didn't make it in the last summer school. I was sitting in I think I was sitting in the modeling session. So
you could join the breakout groups too if you
actually no. Yeah, the first day I was accidentally put into a breakout group, and now they don't let that happen anymore.
We've learned our lesson.
voted off the island. All
I think I was in your very first one of these, Eric, but I don't remember it at all.
No, it's deep
Sorry.
in your subconscious now.
I think you tortured me for a lot longer than just that.
Yeah, you got the worst of it. But you actually made a huge change in my understanding of writing.
Oh,
you are
God.
responsible for actually a breakthrough. You probably don't remember, but I had a breakthrough right there in your text.
Okay. I don't know.
Changed my life.
Great. Thanks. I don't think that's a compliment, actually.
No, it is.
Okay.
Yeah, and it's reflected in everything I'm going to say later. But I don't attribute it to you, so.
Although it is recording now, so everyone will.
So it's there for posterity. Excellent.
I can clip that little section if you'd like, Krista.
It's okay.
Put it on the front page.
Yeah.
Okay, how are we doing?
One more minute. Okay, I'm going to start bringing them back now.
Great. Okay, welcome back. All right, so my question to you guys is, was there consensus or was there a lot of diversity of opinion? Did you guys all agree? Disagree?
I think we mostly agreed. We didn't get to the conflict phase.
Okay,
of it.
no, that's fine, that's
There was
fine.
no
And what did
time.
you agree? What did you agree on?
That co-authors and authors should actually contribute. And we also talked about the balance between the acknowledgement versus being a co-author, like where is that? And then mentioned credit, like the credit author statement that have been introduced lately. So if you don't fit any of those boxes, maybe you shouldn't be a co-author.
Yeah, that's a really good point. Those co-authorship attributions are a new thing in journals, and they're really helpful in objectively assessing this. Paul, can you turn on the slides there? So is that common to most of the groups? Did you cover the same sort of ground? So I was just going to show you one thing on this subject. That is, people have thought about this for a long time, and various groups that have looked into scientific ethics and scientific standards have commented on this. And there are resources you can go to if you want to understand what the scientific community best practices are, and that you're not like stuck on an island having to make these decisions, sometimes with senior scientists, and it can be difficult for young scientists. So I just picked one here. This was from the German DFG. It's a little bit old, 1998, but this was from a publication on regulation of science, which is really important because for the public to have trust in science, we have to, adhere to a certain set of standards and best practices, and that's super important in all fields. And so this was their statement about the criteria for authorship, and they also outlined a specific set of comments of things that don't warrant co-authorship, and they're interesting. Things like responsibility for obtaining the funds does not warrant co-authorship, although the person who's responsible for the funds may very well have been a person who conceived the ideas. but it's the ideas that would then, warrant the co-authorship. And certainly the last one, directing an institution, is not a justification for a co-authorship. And this used to be a big problem in science, where there was sort of an institutional hierarchy and people expected to be co-authors on things from an institution. Anyway, there are more recent and many different versions of this that you can see. Lucas, question.
What do you mean with contribution of important materials? Does this include figures or what's?
No, let's say that you made some measurements on some particular rocks and somebody gave you those rocks.
Okay.
Right? It could be anything like that, water samples or something like that. Just the giving of samples is not in and of itself an intellectual contribution warranting co-authorship. I think that's what they're trying to say. Yeah. Okay, so just something to think about. And the acknowledgements have become devalued over the years. People don't count them in promotions and tenure and things like that, but they are a very important thing. And often I'm happier about seeing somebody thank me for something I actually did than I am seeing my name on some paper that I had nothing to do with. So don't forget to acknowledge people. It's much appreciated. Okay. Okay, so now I want to talk about my approach to writing that I call it sort of meta writing. And maybe that will make sense to you, maybe it won't, but I want to step through it and see what you think. So first off, let me say what's different about professional writing from every other kind of writing that you're going to do. Okay, what's different is professional writing has a purpose. There's somebody on the other end that you need to deliver information to, and they need to get that information quickly, right? We're all trying to read papers, we're all trying to learn and advance our science. We're not reading these things like novels where we stay up late at night and read and enjoy ourselves. This is for professional purposes. And so how do we accomplish that? What's the most important ingredient? That is that the information has to be packaged in the way they expect to find it. Okay, that's what makes it an efficient transmission of information from you to this reader. Okay, and in doing that, organization is everything, right? So I start from the perspective of this meta, step back a little bit from the information and the content and the science and all the questions and think about the organization, okay? Think about the structure of this thing I'm writing and the purpose of it, okay? Now, the document might be a journal article. That's what we're kind of going to talk about mostly here, but it also could be your dissertation, right? It could be, this is a scalable approach to writing, okay? Scalable, yeah. So any questions about that? Does that make sense? And whether it's meta or not, that's a little tough because the word meta is changing, right? Now meta means like you're off in some virtual headset or something like that. I'm not talking about virtual writing. I'm just talking about stepping back from the content. Okay, so let's talk about a journal article, all right? I would argue that you've got to pick a vehicle that you're going to use to push this information out to your ultimate receiver. So what's that vehicle? In today's world, it's a journal. What journal are you going to pick? Students used to ask me, should I pick the journal before I write the paper, or should I write the paper and then pick the journal, right? So I would advocate always pick the vehicle and you're going to write to the vehicle. So how do you pick a journal? That's my question to you. How do you pick a journal? Anybody? There's two ways. Can anyone tell me one? Well, there are more than two ways, some good and some not so good. Go ahead, Lee, you're going to say something or?
Not really, but there's, for a specific field, there's only that many journals that fit your work, right? like
I'm not sure what that means. So what is the criteria to say, you say oh, there's only a few journals, say 5 journals. What do you mean there's only 5 journals? How do you know there's only 5? Based on
I
what?
mean, those top journals that you know you read similar work that have been published on those journals.
Okay, so subject-driven decision.
Yeah, yes.
I'm
yes.
writing about biogeochemistry, so I'm only going to publish in biogeochemistry journals. Okay, that's
Yes.
one way. That's fine. What's another way? Maybe you haven't thought about this. So another way is to say readership. Maybe I'm doing biogeochemistry, but I want molecular biologists to read my paper. I want them to understand what's a really important problem in biochemistry that's related to biogeochemistry, but their community is not working on these problems and they're interesting for them. So you might decide I'm going to publish my paper in one of their journals in order to reach that audience. So the first was subject-driven choice of journals. Second one might be audience-driven choice of journals. It might be a challenge to do it, but you could do that. You could write a paper on physics aimed at physical chemists. You could write a paper on chemistry aimed at chemical physicists, so forth. So you got to decide who you want to reach. We're going to format to the journal requirements. That's a no-brainer because that's where we want this thing to end up. There's no point writing it in some different format and then reformatting it. We're going to define the sections and exactly what information should be in which section. The sections are often prescribed by the journal, but they're totally generic. You need an introduction, you need a methods, you need a result, but they don't tell you anything about what goes into those sections, or very vaguely. What I'm advocating here in number 4 is you're going to decide exactly what information goes in each section, and you're going to define down to the paragraph level what you're going to deliver to these readers. Okay? And you're going to do that before you write the paper. Beyond that, we're actually going to define the first sentence of each paragraph, and we're going to try and enforce paragraph scope. And then finally, when we've produced this document that exactly meets our purpose and the readers we want to reach, we're going to review the whole thing. We're going to look at the terminology we used, make sure it's appropriate, and make sure it's consistent, that we don't change terminology in the middle of our paper, or we're not using some outmoded term for something. So we will review that terminology and simplify our grammar. Okay, so to me, these are the basic steps. It works in a scalable way, and it makes sense to me. Now, whether it makes sense to you or not, I don't know. Questions? Okay. Does anybody write this way?
Can I say something?
Yeah.
Because I think I never thought it in this way, but it looks like kind of like boxes that are already in this format, and you put like the information in.
Yes. So yes, you could imagine a template, a computer template, right? And that's essentially what we're doing. Okay, and I'll show you in a little bit more detail how this works.
Okay.
Let's go. Okay, so here's an example of a paper I want to write. And I'm going to do a meta writing of this paper. Okay, I'm not writing the actual paper yet. I'm writing what the paper is going to be like. So I'm going to have an introduction. It's going to have a bold heading that says introduction, and it's going to have 3 paragraphs. It's going to have one that explains the science question. What is it that motivated me to get going on this, right? If climate is changing, we need new science to define how climate's going to change on 100-year timescales. That's turned out to be a huge challenge for our field. And that's because we don't really understand the carbon cycle and blah, blah, blah. That's the level. What is the science question that's driving you to do this work? 2, what's been done on this? Why is this such a hard problem, right? There's been some breakthroughs over the years, but clearly And we want to show people that we know the work that's been done and give people credit for the discoveries that they've made and the breakthroughs. But sometimes there are things in the field that disagree. There are maybe two ideas that have come down through the literature, and we want to highlight that there's differences of opinion about certain important things. Who knows? That's all goes in paragraph 2. That's where you get to show that you are aware of what's been done up to this time, and you help the reader to get there. And then there's paragraph 3. What have I done? Why did I do this? So the specific approach for your study, the goals for this study, why it's novel or not novel. Maybe you're just repeating something because it needs to be repeated. That's fine. The audience needs to know that. And this is a place where you get to kind of define what the story is going to be of this paper. If you can't do that, you're going to have a heck of a hard time writing this paper. Okay. So, fine. That's methods. If I can get through that done, great. If I can't get through that third paragraph, or even the first paragraph, but definitely the third, then I better stop and settle down and have some discussions with my co-authors and colleagues and try to figure out what this paper is really going to be about. Okay. So that's the meta-analysis. We haven't said anything specific about our science. We haven't gotten into the weeds. We're not worried about our grammar. We're just focused on the big picture. You can do the same sort of analysis of the method section. Okay. So I'm an experimentalist, you know, with knobs and gizmos and measuring things. So, you know, I tend to think about my methodological strategy. So in this paper, you know, I'm going to use direct shipboard observations to try to measure these quantities. And I'm going to use this technique of mass spectrometry and make these measurements and calculate fluxes. So I give a little explain what I'm really trying to do methodologically. Maybe it would be different for a modeler. Then I got to write a paragraph saying what's the instrumentation I'm using so they understand what this is. Maybe it's a model that I'm using, and so I want to describe the resolution and the physics that's in this model, whatever. Then I need to discuss the procedures. It's not enough to just talk about a hunk of metal. It's how did we do this? What are the procedures? What flow rates did we use? What blah, 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 voltages, this or that. And then finally, what was the performance of this system under the conditions that we're going to be telling you the results about? So for me, it would be what's the precision and accuracy under real world conditions? And how do I measure the precision and accuracy? What's important for the reader to know about how you did this work in order that they could repeat it someday and check their performance against yours? Models have the same issues, data analysis, satellites. It can all be applied, the same sort of stepping back and thinking about what does somebody need who wants to repeat this work 20 years from now or 50 years from now. Any questions about that? So you have to tailor it to your kind of work. Okay. So I'm a fan of results and discussion rather than just results, because I think it gets pretty dry when you just talk about results. And then as a reader, I forget what the results were when I get to the discussion. So I'm like lost and floundering around. So I tend to combine these two, but you don't have to. So how do you do this? And I'm just giving an example. Every paper is different. But for me, it's one big result. So I made, let's say I'm measuring chemicals in an ice core. I made 500 measurements of this thing. They had this mean and this variance and this trend, and there's a really big thing in the middle, and it's all very, very interesting. I'll give some statistics. So describe the result. There's usually a big figure that goes with this. But you're writing about the contents, telling the author telling the reader what to look at in that figure. So in other words, you know, the trend of methane is up, and it's gone up by 20 parts per billion in the last 5 years. Parentheses, figure 1, and there you go. That's fine. Maybe you want to write a paragraph comparing the results to comparable data from another study. Maybe some other lab just measured the same trend and you get to compare yours to their trend and everything's wonderful, or this is really different from what other people saw. Maybe you want to write a paragraph on synthesis. So let's say, knowing this now, when we look at what else has been done, we learn something new. We learned that the northern hemisphere has been going up, but the southern hemisphere has been going down. That's really interesting and unusual and surprising, and maybe there's a new figure that highlights that at a synthesis level. Not necessary to have all of these, and they have to match yours, but maybe you have 4 or 5 different results in your study, and you need to do this for 3 or 4 different things. You know, it's a little hard to generalize for everything, but that's the standing back and looking at the big picture. Okay, how many items do I have to talk about? And have I conveyed this information? Questions? Okay, let's keep going. So sometimes you want to have a conclusion section. I am super big on conclusions. And I want to emphasize that conclusions and summary are different things. Okay, a summary is a rehashing of the paper that you're writing. Conclusions is a step back, providing new insight and information to that reader. I think conclusions are super important. That's what I go and read. And I am almost universally disappointed when I get there, because what I find is a summary. And I already know the summary because I've looked at the figures and I already know what they did. What I want to know is, what do they think about what they did? Right? So in other words, you could present your new biogeochemistry cruise and your new data, and you could decide that, you know what, this was really the wrong thing to do. Like, this was a great project and we got everything we went for, but it turns out, now that we think about it, this was the wrong thing. We should have gone in the other season. We really need to do this again. Well, that's a higher level of understanding that the audience really needs, okay? So you've worked on this stuff a lot and you've thought about it a lot, and you have a lot of higher order thinking that's of value to the audience, and they deserve that. They want that. That's really where the value is, okay? So I said paragraph 1, maybe you restate the major finding, but do it succinctly. This is not a time to rehash everything. Was it expected or unexpected? Did it confirm or disrupt your prior view of something? Or are there major limitations? Like, we set out to do this, we did it, now we realize it doesn't prove what we thought it would. If it had turned out that way, it might have proved something, but it turned out this way and it doesn't prove what we thought. Then that could be enough. You could be perfectly happy with that, is plenty. But I would also advise that you tie back to paragraph 1 at the beginning of the introduction, which is the reason you got into this in the first place. Maybe it's the bigger global change problem or ocean acidification or something. So where does this study fit into that? Did we move the ball forward on the big problem? Did we open a path for how to do this better in the future? You know, give us some context there. Maybe it guides us towards what we should do next, okay? So I said homework here. This is not real homework. This is just for you to think about in your spare time when you are stuck and can't figure out what to write, is pull out 2 or 3 journal publications that you really admire, or that you've used, and look and do the meta-analysis on them. Oh, what has this author done in paragraph 1 of the introduction? What did they do in paragraph 2? What did they do in paragraph 3? How did they structure their results and discussion? Is there a structure to it, or is it just some meandering mess? Chances are the papers that are meandering messes are not high on your list of my favorite papers. And I think what you'll find is there's some real commonalities to good papers from this level of analysis. Any questions? Okay. Let's charge on. Okay. So next comes the harder part. We've laid out our paper. We have a roadmap. We know where it begins and ends. now it's time to bring some discipline. And so I'll start with the paragraph level. Okay, so you know what these paragraphs are for. Now you just got to sit down and write them. And after you've written it, of course, you should read it. And then I want you to ask this question. At the paragraph level, is it immediately obvious from the first sentence what the paragraph is about? In other words, could the reader tell you the meta purpose of the paragraph by the time they finish the first sentence? Okay, so if my paragraph is prior work on this field, your first sentence of that paragraph should be the most important prior work on this field was. Okay, or a ton of prior work has been done in these aspects of this problem. Okay, so this is professional writing. We're helping our reader understand why they're reading this paragraph and what they're going to read in the paragraph. And then I say, does every sentence help achieve the specific purpose of this paragraph? If it doesn't, then throw it out. Or decide, you know what? That sentence has such an important idea. I really think it's important for my reader to understand that, but it's not exactly the purpose of this paragraph. You're the author. If you want to write another paragraph about that thing, go right ahead, right? You're in control of this thing. But don't jam it into another paragraph as sort of an added bonus. Oh, my reader will love this. I'll just slam this in the middle. Think how happy they'll be. Okay, so there you go. Right, so I wrote a paragraph for you guys. How do you like that? That's like going above and beyond. I made one up for a paragraph 3 of the introduction. So I'll let you read it.
Hey, Eric, while the students are reading, I think we probably need to wrap it up in the next minute or two, just so we can finish up with the posters.
Yep.
Thanks.
Okay, I'm going to leave that there. You can look at it later. It's fine. It's just a paragraph. It does the job and it's done. You don't have to write 10 more sentences. If it does the job, it's done the job. Okay, we got to bring this home. Discipline at the sentence level. Do your sentences have a subject, a verb, and an object, and one of each, not 5 of each or 7 of each? Is the second one is really important to me, close to my heart. Is the intended subject of the sentence the actual grammatical subject? And I'll show you something in 1 minute to describe that. Are you using the right terminology? And do you have words like it that absolutely baffle your readers and force them to reread the paragraph to find out what you're talking about? All right, so here's my 1 minute exercise. Oops. Okay, I just want you to look at first sentence here. Many research studies have shown that climate is very sensitive to the levels of CO2 in the atmosphere. It's a lovely sentence, but what's wrong with it? What's the subject of the sentence?
Many
The grammatical
studies is the
subject.
subject.
Go ahead. Is what?
Many studies is the subject, but I think it should
Yes.
be like the high levels of CO2 in the atmosphere.
Very good. Correct. So many studies is of no use to anybody. This wasn't the paper about how many studies there have been. It's a paper about climate. So you want to make climate the subject of your sentence. So after you've organized your paper and after you've written all these paragraphs, go back down and impose discipline at the paragraph level and then at the sentence level. Okay, now Katya's going to kill me, but I'm going to show you the last. Okay, so this is it. This is the whole thing wrapped up in a nutshell. From large scale, zooming to small scale, summarizing everything we just talked about. If you can do this, you can write anything, whether it's 10 pages or 200.
Thanks, Eric. This is great. I actually am really enjoying this myself. I'm thinking I need to get back to that paper that I've been ignoring. So thanks very much. And thanks to Vero as well. That was an excellent practical session. So we're not going to have breaks since you all already had one. We're going to move right on to the poster session. So thanks everyone for their engagement so far in the session. This is the last one. Everyone throughout the course of the week will have a chance to present their posters. And don't forget that there's 2 poster prizes per day. So everyone will have the chance to do an informal 1-minute flash presentation on their poster. So be ready to go one after the other. It's pretty fast paced. We'll just keep going. I know you guys have already done this a couple days in a row. So why don't we go ahead and get started?
Hello, everyone. It's a pleasure to be here with you. Well, in my postdoctoral position, my aim was to study the Pacific of Mexico and identify what driver changed the PCO2 in different scales, spatial and temporal. And we find that the highest values of PCO2 were in spring, in the latitudes higher than 21, and in the Gulf of Tehuantepec, the dynamic is very different in Tehuana's winds, that is inertial winds in the season October to March is very intense and is very important driver in this zone. And also we identify that with PCO2 and FCO2 fluxes, we can regionalize and to do section in the Pacific of Mexico in 25 years of this study. Thank you.
Hello, everyone. Okay, so we went on a cruise on the Mexican Pacific waters, and just to do some research on the Mexican oxygen minimum zone. And we took some discrete samples to measure the surface carbon dioxide fluxes. And what we found was very interesting because we found some points with high surface PCO2, and if you translate these values of PCO2 to carbon fluxes, we found that the study area of the Mexican Pacific waters behaved as a weak source of carbon dioxide to the atmosphere, with values between 2.4 to 7 millimoles of carbon per meter per day. And all of this was due to the physical features of the area, speaking of the wind, because we found on those days, I know that the ocean is not a static system, but we can take some snapshots of the ocean. And we found that on the days of the cruise, we had high winds and a positive rotational wind, wind stress field.
Thanks very much.
Well, usually sensors used to quantify PCO2 are very expensive, are around $5,000. So makes them inaccessible to many laboratories. We aimed to make an inexpensive PCO2 sensor that could be obtained easily and relatively quickly. After obtaining a sensor from Amazon, we looked for a way to generate a replicable measurement method, which could be applied for scientific or educational purposes. We use an Arduino Uno microcontroller to integrate the PCO2 sensor with a humidity and temperature sensor and add a data logger that stores everything on a micro SD card. It was found that with 2 gases of known concentration, it was possible to have an accuracy of about 20 parts per million. And that's it.
Thanks.
Hello, everyone. I'm Masha Hussain from the University of Illinois. I believe that today's session has informed us all about the Upwelling region, which makes my task easier. I have recently started working on the Benguela Upwelling region with the aim to improving the marine aerosol representation in the 3D climate model, which I'm using right now called GeoScam. We all know by now that for the projection of the global environmental trends, it is critical to improve our understanding of the ocean atmosphere feedbacks. To this end, I have updated the DMS chemistry of the model by accounting the pathway for the loss of HPMTF to the clouds, which leads to higher sulfur emissions during December. We also look forward to validate the model with recent international field campaigns to improve the parameterization of the ocean atmosphere links. And yes, we are still a work in progress, so any suggestions are highly welcome. Please do visit my poster to know more about my work. Thank you.
Hi, I am presenting on some research that I did during my time working with the National Oceanic and Atmospheric Administration during my undergrad time. This work is focused on the fluorescence measurements related to chlorophyll A and just primary production that was happening. The work was done via some sail drones in the Chukchi and Bering Seas during 2019. And from those, we used fluorescence sensors to measure the amount of chlorophyll that was located. Some interesting findings were the fact that really near ice events, which are on these top 2 figures there, located with the gray background bars, did not actually see any increased chlorophyll production on the surface, which was something that was a little unexpected. All in all, through a variety of different calculations, we were able to compute what the net primary production was, and that's shown on that bottom chart there. Interestingly enough, the highest primary production was found in the green belt, a little bit lower, and less production occurred in the higher Arctic regions during this study. Thank you.
Hi, you all. This is Lei from SUNY ESF. My poster is about this organosulfur compound DMSP, which has been extensively studied for about 4 decades due to its important role in global sulfur and carbon cycle and in climate. Surprisingly, very little is known about how DMSP and its degradation product accurately behave in coral reef ecosystems. Although we know coral reefs hold a large quantity of these compounds. In this study, we found that DMSP and acrylic are ubiquitous in the reef water of the island of Maria in French Polynesia, with reef benthic organisms as important producers. And also the biological consumption of these compounds is extremely fast in the reef, suggesting that these compounds play an important role as biological substrates for the reef microorganisms. Feel free to reach me if you have any questions. Thank you.
Hello, hello everyone. My name is Thais from the National Institute for Space Research in Brazil, and today I present my ongoing work, which is entitled Direct Observations of Ocean Atmosphere Heat Fluxes in the Western Tropical Atlantic. In this work, we analyzed data collected during a campaign of the Brazilian branch of the Pirata project with the objective to evaluate the heat fluxes between ocean and the atmosphere and the position of the intertropical convergence zone in the Western Tropical Atlantic. Till this point, we are able to identify the position of the ATCZ between 6 and 10 degrees north, The results also indicate that the Western tropical Atlantic was a source of heat to the atmosphere during the study period. The difference between the bulk estimates and the actual variance methods are still being evaluated, but they suggest that there is a need to better understand the bulk variables in this region in order to improve the efficiency of these parameterizations. Thank you for your attention, and if you have any questions, please contact me.
Hello, everyone. I'm Raíza Chieli. I'm a PhD candidate at the Federal University of Ceará in Brazil. My research is in the context of climate change and how it promoted more studies in marine and aquatic systems and their roles in the carbon cycle. However, these two areas are still neglected in budgets and models, especially tropical estuaries. So our study area is a large river delta, the Parnaíba River Delta. That's the largest open delta in Americas, and it's a delta which is dominated by mangrove forests with little human interference, and therefore it's a key environment in understanding the changing of carbon cycles in natural systems. So our main goal is to understand the spatial and seasonal variability of CO2 and its fluxes in this unique place, and also how the origin of the organic matter is related to this variability. So if you guys have any questions, you can send me a message or contact me via the Wova platform. Thank you.
Hello, everyone. I am Juan Camilo Torres. I am a PhD candidate at the Federal University of Rio Grande in Brazil. And this is my PhD project, focused on the effect of the climatic modes on the cryosphere on the Peninsula Antarctica and how the increase in meltwater fluxes can modify the carbon dioxide fluxes while physically, but more changing the physical issues of the surface of the mixed layer, and biogeochemically how the input of the solvent in organic iron from the cryosphere, from the shade of the subsurface, so the dust over the glaciers, over the ice, can modify the reasons of dissolved inorganic micronutrients in the region of the North Atlantic Peninsula, and especially in the Brainfield Strait. This is based on work on over the, I'm sorry, I just forget the name. BSOS is a ocean estimation state that brings the parameters of changes of water masses and biogeochemical parameters. Thank you.
Thank you.
Hello, I'm Josefa Verdugo. I'm working at the Biogeochemical Laboratory at the University of Concepcion in Chile. And with this project, we aim to understand and to determine the effect of the freshwater layer that is formed by glacier melting and continental runoff on the exchange of methane between the ocean and the atmosphere in fjords and channels of northern and southern Patagonia. And in addition, we want to make experiments to determine the fraction of methane that is lost by aerobic methane oxidation associated with different stratification levels in this zone in southern Patagonia. So if you have questions or if you have interest, I'm happy to talk more about this. So thank you.
Hi, I'm Hashan Kokuhenendige. I'm a PhD student at University of Southampton. It's a great opportunity for me to participate in this year's SOLAS Summer School and share what I'm looking in my PhD research. My study focuses to understand the effect of various oceanic processes and anthropogenic influences on the marine carbon cycle, particularly in the Northeast and Southeast Atlantic Ocean, using stable carbon isotopes in marine sample. So why it is important to study carbon cycle? Marine carbon cycle play a vital role in determining the biogeochemistry in marine environment. So that ultimately regulates the biodiversity and the productivity. So however, these carbon cycle is influenced by various oceanic processes as well as the man-made influences since the beginning of industrialization. So these make the carbon cycle more complex. So that's why we use stable carbon isotopes, because each carbon pools are characterized by a unique isotopic signatures. So they are, because of that, they are very powerful traces to understand the effect of various processes acting on carbon cycle. So in that way, that will help us to illustrate the current carbon cycle in more detail. So I hope my research will support the Sustainable Development Goals, especially 14 and 13. Thank you.
Thanks very much. Well done, everyone. That's a really interesting group of posters. Thanks for that. Unfortunately, we ran over a few minutes, so we don't have time for discussion this evening, but of course we can use Hoover and the Hoover platform to have conversations around posters and ask questions, send direct messages to people if needs be. And if there's any technical questions, you can always email Paul and Becky, but that wraps it up for today. So thank you so much for your time and participation. Remember, we start up again tomorrow from 7 to 11:30 UTC, and that is the last session. So I look forward to seeing all of you tomorrow morning. Have a good evening, afternoon, morning, day. See you all tomorrow.
Bye-bye.
Bye.
Bye-bye.